ओम अनुमते अनुमन्यस्व। ओम स्वस्त्यनुमन्यस्व। ओम देवसवितः प्रसुवयज्ञं प्रसुवयज्ञपतिं भगाय दिव्यो गन्धर्वः केतपुकेतं नः पुनातु वाचस्पतिर्वाचं नः। नहीं करो, बाकी सब मत। ओम अग्नये स्वाहा। इदमग्नये इदन्नमः। ओम सोमाय स्वाहा। इदं सोमाय इदन्नमः। ओम प्रजापतये स्वाहा। इदं प्रजापतये इदन्नमः। ओम इंद्राय स्वाहा। इदमिन्द्राय इदन्नमः। सामग्री की आहुति। ओम सूर्यो ज्योतिः ज्योतिः सूर्यः स्वाहा। सूर्यो वर्चो ज्योतिर्वर्चः स्वाहा। ओम ज्योतिः सूर्यः सूर्यो ज्योतिः स्वाहा। ओम सजुर्देवेन सवित्रा सजुरुससेन्द्रवत्या जुषानः सूर्यो वेतुः स्वाहा। ओम अग्निर्ज्योतिर्ज्योतिरग्निः स्वाहा। अग्निर्वर्चो ज्योतिर्वर्चः स्वाहा। ओम स्वाहा। सजूरात्रेन्द्रवत्ता जुषानो अग्निर्वेतुः स्वाहा। भूरग्नये प्राणाय स्वाहा। इदमग्नये प्राणाय इदन्नमः। ओम भुवर्वायवे पानाय स्वाहा। इदं वायवे पानाय इदन्नमः। ओम सुरादित्याय व्यानाय स्वाहा। इदमादित्याय व्यानाय इदन्नमः। ओम भुवः स्वरग्निर्वायवादित्येभ्यः प्राणापानव्यानेभ्यः स्वाहा। इदं वायवादित्येभ्यः प्राणापानव्यानेभ्यः इदन्नमः। ओम आपो ज्योतिरसो अमृतं ब्रह्मभूर्भुवः स्वरों स्वाहा। ओम यां मेधां देवगणाः पितरश्चोपासते तया मध्यमेधयाग्ने मेधाविनं कुरु स्वाहा। ओम स्वानि देव सवितर्दुरितानि परासुव यद्भद्रं तन्न आसुव स्वाहा। ओम अग्नेनाय सुपथाराय अस्मान् विश्वानि देवस्मज्जुहुराणमेनो भूयिष्ठान्ते नम उक्तिं विधेम स्वाहा। ओम भूर्भुवः स्वः तत्सवितुर्वरेण्यं भर्गो देवस्य धीमहि धियो यो नः प्रचोदयात् स्वाहा। ओम भूर्भुवः स्वः तत्सवितुर्वरेण्यं भर्गो देवस्य धीमहि धियो यो नः प्रचोदयात् स्वाहा। ओम भूर्भुवः स्वः तत्सवितुर्वरेण्यं भर्गो देवस्य धीमहि धियो यो नः प्रचोदयात् स्वाहा। केवल घी की आहुतियां होंगी। ओम भूर स्वः तत्सवितुर्वरेण्यं भर्गो देवस्य धीमहि धियो यो नः प्रचोदयात् भुवः स्वः सवितुर्वरेण्यं भर्गो देवस्य धीमहि धियो यो नः प्रचोदयात् भूर्भुवः स्वः तत्सवितुर्वरेण्यं भर्गो देवस्य धीमहि धियो यो नः प्रचोदयात् स्वाहा। केवल घी की भूर्भुवः स्वः तत्सवितुर्वरेण्यं भर्गो देवस्य धीमहि धियो यो नः प्रचोदयात् स्वाहा। ॐ भूर्भुवः स्वः तत्सवितुर्वरेण्यं भर्गो देवस्य धीमहि धियो यो नः प्रचोदयात् स्वाहा। ॐ भूर्भुवः स्वः तत्सवितुर्वरेण्यं भर्गो देवस्य धीमहि धियो यो नः प्रचोदयात् स्वाहा। केवल घी की आहुतियां होंगी। ओम भूरग्नये स्वाहा इदमग्नये इदन्नमः। ओम भुवर्वायवे स्वाहा इदं वायवे इदन्नमः। ओम स्वरादित्याय स्वाहा इदमादित्याय इदन्नमः। ओम भूर्भुवः स्वरग्निर्वायवादित्येभ्यः स्वाहा इदमग्ने वायवादित्येभ्यः इदन्नमः। अब उत्कृष्टकृत आहुति होगी। यदस्य कर्मणो अत्यरीरिचं यद्वान्यो न मिहाकरम् अग्निष्टसृष्टकृतविद्यात् सर्वं संसूतं करोतु मे। अग्नये सृष्टकृते सुहूतहुते सर्वप्रायश्चित्ताहुतीनां कामानां समर्दयित्रे सर्वान्नः कामान् समर्दय स्वाहा। इदमग्नये सृष्टकृते इदन्नमः। ॐ स्वाहा इदं प्रजापतये इदन्नमः। ॐ भूर्भुवः स्वः अग्न आयुंषि पवस आसुवोर्जमिषं च नः आरेबादस्वदुच्छुनां स्वाहा। इदमग्नये पवमानाय इदन्नमः। ॐ भूर्भुवः स्वः अग्निरिषि पवमानः पाञ्चजन्यः पुरोहितः समीमहे महागयं स्वाहा। इदमग्नये पवमानाय इदन्नमः। ॐ भूर्भुवः स्वः अग्ने पवस्व स्वपा अस्मै वर्चः सुवीर्यं ददद्रयिं मयि पोषम्। इदमग्नये पवमानाय इदन्नमः। ॐ भूर्भुवः स्वः प्रजापतेन त्वदेतानन्यो विश्वा जातानि परिता बभूव यत्कामास्ते जुहुमस्तन्नो अस्तु वयं कामपतयोरयीनां स्वाहा। इदं प्रजापतये इदन्नमः। सामग्री की भी आओ। तन्नो अग्ने वरुणस्य विद्वान् देवस्य हेडो अवयासि शीर्षाः यजिष्ठो वह्नितमः सुचानो विश्वा देशांसि प्रमुद्यस्मत् स्वाहा। इदमग्नि वरुणाभ्याम् इदन्नमम ॐ सत्त्वं नो अग्ने वमो भवोति नेदिष्टो अस्य उषसौ व्युष्टौ अवयक्षनो वरुणं दराणो विहि मृडीकं सुहवो न एधि स्वाहा इदमग्नि वरुणाभ्याम् इदन्नमम ॐ कामवत्युराजके नमः ॐ तत्त्वायामि ब्रह्मणा वन्दमानस्तदा शास्त्रे यजमानो हविर्भिः अहेडमानो वरुणे हवोद्युरुषं समान आयुः प्रमोषि स्वाहा इदं वरुणाय इदं नमः ॐ ये कथं वरुण ये सहस्रं यज्ञाः पाशा वितता महान्त ऋणो अद्य सवितोत विष्णुर्विश्वे मुञ्चन्तु मरुतः स्वर्गः स्वाहा इदं वरुणाय सवित्रे विष्णवे विश्वेभ्यो देवेभ्यो मरुद्भ्यः स्वर्गेभ्यः इदं नमः ॐ अयाष्टाग्ने अस्य नारिषष्टिपाश्च सत्यमित्वमया असि अयानो यज्ञं वहास्ययानो देहि भेषजं स्वाहा इदमग्ने अयसे ॐ उदुत्तमं वरुणपाशमदवादमं विमध्यमं क्षथाय अथा वयमादित्यव्रते तवानागसौ अदितै स्याम स्वाहा इदं वरुणायादित्याय दितये च इदं नमः ॐ भवतं नः समसौ सचेतसा वरेपसौ मा यज्ञोहिं सृष्टं मा यज्ञपतिं जातवेदसौ शिवौ भवतमद्यनः आहा इदं जातवेदोभ्याम् इदं नमः ॐ सर्वं वै पूर्णं स्वाहा सर्वं वै पूर्णं स्वाहा सर्वं वै पूर्णं स्वाहा अग्नये स्वाहा अग्निशोमाभ्यां स्वाहा विष्णवे स्वाहा रूप प्रभो हमारे भाव उज्जवल देवे छल कपट को मानसिक बल दीजिए को ले रिचाए सत्य को धारण करें हर्ष में हो मग्न सारे सागर से तरे अश्वादिक रचाए यज्ञ पर उपकार को धर्म मर्यादा चला कर लाभ दे संसार को सदा भक्ति से यज्ञादि हम करते कहीं भोग पीड़ित ताप सब हरते कहीं भावना मिट जा हमसे आप अत्याचार अपनाए पूर्ण होवे यज्ञ से नर नार की लाभकारी हो हवन हर प्राणधारी वायु जल सर्वत्र हो शुभ गंध को धारण किए स्वार्थ भाव मिटे हमारा प्रेम पथ विस्तार हो मम का सार तक प्रत्येक में व्यवहार हो हाथ जोड़ झुकाए मस्तक वंदना हम कर रहे नाथ करुणा रूप करुणा आपकी सब पर रहे। पूजनीय प्रभु हमारे भाव उज्ज्वल की। छोड़ देवे छल कपट को मानसिक बल दीजिए। प्रभु आत्मिक बल दीजिए। भवन्तु सुखिनः सर्वे सन्तु निरामयाः। सर्वे भद्राणि पश्यन्तु मा कश्चित् दुःखभाग् भवेत्। सबका भला करो भगवान, सब पर दया करो दयावान। सबको दो वेदों का ज्ञान, सबका सब विद हो कल्याण। त्वमेव माता च पिता त्वमेव, त्वमेव बन्धु च सखा त्वमेव। त्वमेव विद्या द्रविणं त्वमेव, सर्वं मम देव देव। सर्वं मम देव देव। साथ बोलेंगे ओम सत्याः सन्तु यजमानस्य कामाः पूर्णाः सन्तु शुभाः सन्तु शुभाः सन्तु मंगलाः सन्तु यजमानस्य कामाः। स्वस्ति स्वस्ति स्वस्ति। ओम स्वस्ति न इंद्रो वृद्धश्रवाः, स्वस्ति नः पूषा विश्ववेदाः, स्वस्ति नस्तार्क्ष्यो अरिष्टनेमिः, स्वस्ति नो बृहस्पतिर्दधातु। ओम भद्रं कर्णेभिः शृणुयाम देवा, भद्रं पश्येमाक्षभिर्यजत्राः, स्थिरैरङ्गैः सुष्ठुवागुंसस्तनूभिर्व्यसे महि देवहितं यदायु। ओम उत्तिष्ठ ब्रह्मणस्पते देवान् यज्ञेन बोधय, आयुः प्राणं प्रजां पशुं कीर्तिं यजमानं च वर्धय। ओम स्वस्ति ओम स्वस्ति। कार्यक्रम 2 दिन का तिथि है। आपका स्वागत है सर इस प्रांगण। सदी पादम सर भाई साहब बहुत अच्छा रहा है सर। आपके मुख्य उपस्थित सभी जवान करेंगे।
हृदय से आभार व्यक्त करूंगा के अवसर पर आपका हृदय से आभार। कि हम सभी लोग जानते हैं कि विद्यालय के माध्यम से पादप कुंभ का आयोजन हो रहा है। कुंभ महाकुंभ के अवसर पर समझता हूँ कि पूरे शहर में पूरे प्रदेश में कार्यक्रम की गूंज लोग पत्र पत्रिकाओं के माध्यम से, सोशल मीडिया के माध्यम से देखते हैं कि कितने अच्छे कार्यक्रम हमारे इस परिसर में हो रहे हैं, इस विश्वविद्यालय में हो रहे हैं। इससे ये विज्ञान के क्षेत्र में एक बहुत पूर्ण कार्य है। आप लोग जानते हैं कि इस कुंभ के साथ साथ 2 कुंभ और हो रहे हैं, एक कुंभ के रूप में और एक पर्यावरण कुंभ के रूप में। और तीसरा ये जो कुंभ हो रहा है, ये पादप कुंभ के रूप में गुरुकुल कांगड़ी विश्वविद्यालय के द्वारा किया जा रहा है। और बहुत सारे कार्यक्रम निरंतर, मैं भले ही इसमें सम्मिलित नहीं हो पाता हूँ, लेकिन इन एक्टिविटीज़ से मैं निरंतर जैसे मेरा संवाद रहता है, तो बहुत अच्छे कार्यक्रम हैं। निश्चित रूप से ये विज्ञान के क्षेत्र में हम लोगों को पुनः अपनी वैदिक ज्ञान की तरफ मोड़ने का एक अभिनव प्रयास है। जैसा कि आप जानते हैं कि हमारे प्रोफेसर दुबे जी और प्रोफेसर हमारे भट्ट जी, दिनेश भट्ट जी, डॉक्टर पुरोहित जी, वैज्ञानिक हैं जिनका अंतर्राष्ट्रीय स्तर पर बहुत अच्छा वैज्ञानिकों के रूप में विभिन्न अपने अपने बहुत बड़ा योगदान है के माध्यम से, और हमारे माननीय कुलपति जी के नेतृत्व में सारी की सारी गुरुकुल कांगड़ी के द्वारा एक ऐसी विधा, वैज्ञानिकों को एक ऐसा मार्गदर्शन मिलेगा कि वो हम आज पुनः वैदिक ज्ञान की तरफ मुड़ेंगे। ज्ञान विज्ञान से आप समझ सकते हैं कि विशिष्ट ज्ञान जो है वेदों में है। और आज के क्षेत्र में यदि मैं आपको बताना चाहूँगा कि जितना भी रिसर्च हो रही है, सभी के सभी प्लांट्स पे ही हो रही है। कहीं भी पूरे विश्व में देख लीजिए, आज केमिकल से वापस वो नेचर की तरफ जा रहे हैं। गुरुकुल कांगड़ी जैसी संस्थाएं एक मार्गदर्शक का रूप दे रही हैं। और मैं इन्हीं शब्दों के साथ आप सभी को इस पादप कुंभ के बधाई देता हूँ। आज साइंस डे के अवसर पर आपको शुभकामनाएं देता हूँ, और अपना एक तरह से आश्वासन देता हूँ कि यदि कहीं भी इस कार्यक्रम में हम लोगों का कोई नुकसान हो सकता है तो मैं अपनी आप लोगों को समझ। आप सभी का हृदय से आभार। नमस्कार, धन्यवाद।
शांति पृथिवी शांति आपः शांति ओषधयः शांति वनस्पतयः शान्तिर्विश्वे स्वाह शांति ब्रह्म शांति सर्वगं शांति शांति एव शांति सामा शांतिरेधि ओम शांति शांति। बोले सो अभय वैदिक धर्म की जय। मर्यादा पुरुषोत्तम श्री रामचंद्र महाराज की, योगीराज श्री कृष्णचंद्र महाराज की, गुरुवर विरजानंद महाराज की, महर्षि देव दयानंद की, कुलपिता स्वामी श्रद्धानंद महाराज की, भारत के सभी श्रेष्ठ व्यक्तियों की, भारत माता की, गौ माता की। शृण्वन्तु विश्वमार्यं वैदिक ध्वनि ओ। वैदिक अभिवादन। आपके आशीर्वाद के बिना बहस करूंगा। सर 2 वर्ड। आज इनॉग्रल है साइंस डे का, तो और अब हमारे प्राध्यापकगण और कई ओकेजन इकट्ठे हो
व्यक्त करते हैं, समाज के लिए दिया आत्ममुक्ति सर हूँ कांगड़ी विश्वविद्यालय है हुआ सदियों से संबंधित बहुत बड़ा काम कर रहा है। पति जी के विश्वविद्यालय के साथ करके मैं बहुत बड़ा हमें का है या कोई भी एकेडमिक वर्क है और रहेंगे विश्वविद्यालय है और दोनों है प्रारंभ से ही की दृष्टि से भी तो मनीषा है, भारतीय मनीषा या कि ये वैदिक या जो अपनी परंपरा स्वामी सिद्धांत बात की बहुत बात का गर्व हो में जिस तरीके से फार्मास्यूटिकल डिपार्टमेंट ने हाल लिया है अपने विकास में और इसकी तरक्की में बहुत ही अनुपम है। ये आशातीत था, कल्पनातीत। हम कभी सोच भी नहीं सकते थे कि ये विभाग के अनुरूप था या नहीं था, वो अलग मैटर है। आज की स्थिति यह है, कल्पना से बढ़कर के काम कर रहा है। एक वाइस चांसलर कह रहा है गुरु कांग्रेस विश्वविद्यालय का श्रेय आप सबको है जो फैकल्टी के यहाँ हैं, श्रेय देता हूँ और साथ में बड़ा श्रेय देता हूँ तो बहुत बड़ा श्रेय देता हूँ प्रोफेसर राजपूत को। मुझे लगता ही नहीं कि राजपूत कब क्या काम कर बैठते हैं, आते हुए बात करते करते हुए मिला कि यू कॉस्ट से और वहाँ से भी ये अनुदान लिया है। जब ये देहरादून जब भी जाते हैं खुद ले आते हैं। बार गए एक मशीन कहीं ना ये भी गाड़ी खोल दो, जो एक जज्बा है काम करने के लिए लोग बहुत से तैयार रहते हैं, लेकिन जो जज्बाती लोग होते हैं वो इतिहास बना, ध्यान रखिएगा। यही मैं देखना चाहता हूँ हर चले कि हर स्टाफ को, हर कर्मचारी के अंदर वो जज्बा हो, वो जज्बा आपको ऊंचा उठाता है। भी ये आलू विभाग देख रहे थे कि मैं तो कच्चा हूँ जी, मैं कच्चा हूँ जी, मैं क्या कर सकता हूँ जी, मैं क्या कर सकता हूँ। कच्चा तो ये भी है, कच्चे तो हम लोग भी हैं। कौन कौन ऐसा है जो पक्का की ये जो एक भाव है हीन भावना का भाव, मैं आप सबसे कहता हूँ, आप जिस हैसियत में जिस कैपेसिटी में है के साथ आपको यहाँ पर अपॉइंट किया है या आप अपॉइंटेड हैं या चयनित है, आपके अंदर प्रतिभा है, गॉड गिफ्ट है, सभी के अंदर होती है। इसको हम सोचना रही है कुछ नहीं आता, उसके अंदर बहुत बड़ी गॉड गिफ्ट है। प्रतिभा का उपयोग करें और जितना करेंगे उतना ही, जैसे बॉल को हम धरती में मारते हैं, जितनी जोर से मारेंगे उतनी ही स्पीड से और उतनी ऊंचाई पर बॉल जाती है, ऊपर जाती है कि नहीं जाती, बताइए विजेंद्र, जाती है कि नहीं? तो जितनी जल्दी जितनी तेजी से इस विभाग के अध्यक्ष आप सब लोगों के द्वारा विकास और उत्थान की बॉल मारी गई, उतनी तेजी से ऊपर गई। आज पूरे देश में ही नहीं दुनिया में आपका मैसेज आता है, हर जो सोशल मीडिया है आपको कवर करता है, आपकी बात कहता है। कितनों के प्रवचन रह जाते हैं उनके अपनी बुद्धि में ही, लेकिन वाणी से निकले
आज देश और दुनिया सुन रही है। सुनिए कि नहीं सुनिए बताओगे आपको। और मुझे इतनी खुशी हुई कि मैं सोच नहीं सकता था यहाँ से एक ना पैसा नहीं लिया, एक ना पैसा नहीं लिया, तो कोई साधन नहीं लिए, विभाग ने साधन भी जोड़ा दिए, यहाँ स्टूडियो भी तैयार कर दिया। बताइए इससे बढ़िया और क्या बात है। बहुत बढ़िया शानदार कभी इंटरव्यू हुई। और एक बड़ा काम किया है कि सभी को ये संदेश दिया कि भाई सब के सब अपनी रिकॉर्डिंग कराएं। डॉक्टर साहब का चिड़िया के ऊपर इतना बड़ा कई बार पता नहीं कितने, जब मैं खोलता हूँ उसमें दिखाई देता है कि चिड़िया के ऊपर आ रहा है, गंगा के ऊपर आ रहा है, पक्षी जो बाहर के पक्षी आ रहे हैं उनके आ रहा है, साइबेरिया से आ रहे हैं, ये पक्षी आ रहे हैं। कहाँ इतनी बड़ा मैसेज विभाग के द्वारा गया, मैं तो बहुत ज्यादा खुश। आपका स्वागत है। किन किन को जोड़ दिया देखो, डॉक्टर साहब भी आ गए हैं आर्मी के, हैदराबाद से आर्मी के ब्रिगेडियर जनरल, जाने कितने लोगों को खड़ा कर दिया केवल मुझे छोड़ करके। ये बता रहा हूँ द्विवेदी का तो वो तो डीन है ना आपके, जब करा दो इनका। तो इन्होंने न जाने किन 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 परिस्थिति में इस छोटी सी उससे उठ करके और आपने एक महान विभाग बना दिया। तो देश में विभाग का एक विश्वविद्यालय का तो आपने विभाग ने भाल किया है, कह सकता हूँ ये हमें गर्व है और ये गर्व का विषय है। आपने बहुत सारी सामने लान जो रहता था, पूरा कबाड़ा था, लग रहा है कि पीछे संडे को छुट्टी में सब विश्वविद्यालय बनता है या जेसी मसीह राजपूत चलवा रहे हैं। तो जो एक जज्बा है वो जज्बा चाहिए। तो स्वामी श्रद्धानंद ने जज्बा यहाँ पैदा किया था, जज्बा मुझे दिखाई देने लगा है। हर विभाग बढ़ सकता है, एक एक विभाग हमारा बहुत नाम रखता है, लेकिन आप लोग नहीं समझते। तो होता है ना, सेब चलता है तो ये सोचता है मैं तो एक सामान्य सा प्राणी हूँ, जिस दिन अपनी दहाड़ मार देता है सारे भाग जाते हैं। तो तब पता लगता है कि मेरे अंदर कितनी पावर है। आप अपनी पावर को इस्तेमाल करें, जगाइए, को जगाएं और बहुत ही सकारात्मक स्वरूप रखते हुए करने वाले आगे चलते हैं। आपको कह देता हूँ, दो भाई हैं, करना और एक कहना। और करना है दो भाई, करने की है एक कमाई, कहना कह के जावे थक, करना पहुंचे मंजिल तक। जो कहते रहते हैं, जो आलोचना करते हैं, कीजिए आप सब लोग, जो आलोचना करते हैं, क्या टिप्पणी करते हैं, करते हैं, सामने बहुत अच्छा बोलते हैं, ये टिप्पणी करते हैं, आलोचना करते हैं, बुराई करते हैं, समझ लीजिए उनका नहीं हो सकता। मैं खड़े होकर रहा हूं, जहां से शपथ ले लो इस वेदी पर, अपने किसी की भी कोई टीका टिप्पणी ऐसे नहीं करें, मानसिकता के प्रतिकूल है, के प्रतिकूल है, व्यक्तित्व के प्रतिकूल है। में बहुत मजा आता है, रस एक बड़ा सुंदर रस लगता है, लेकिन उसका परिणाम है बहुत भयंकर है। किसी की भी मत करो, किसी की भी त्रुटि न निकालो, मैं ये कह दो भाई ये और कर दे हम बहुत अच्छा रहेगा, सहयोग कर दें और ज्यादा अच्छा रहेगा। तो मैं सुनता रहता हूँ विश्वविद्यालय करते हुए चार, कुछ घर की बातें कोई घर की बातें करने में लगे हुए, का और विभाग का भला और अपना भला नहीं, सब निगाह में आते काम करते हैं। पहचान हर जगह होती है। बहुत बढ़िया कहा, कर्मणि कुशलो यस्तु। तो कर्मणि यानी काम में, कुशल माने कुशल, अस्ति माने है। कर्मणि कुशलो यस्तु, जो काम करने में कुशल है तो अपने कुशलता रहे। सबके अंदर कुशलता, ऐसा नहीं है घर में खूब काम करते हैं, पत्नी खूब दौड़ा देती है, लेकिन यहाँ अध्यक्ष कह दे, डीन कह दे नहीं आता साहब मुझे ये कंप्यूटर नहीं आता, और घर में बच्चों को पढ़ाएंगे ऐसे पढ़ बेटा वैसे। ये दोहरा जो चरित्र है बहुत खतरनाक है, ध्यान रखिएगा। बेहद खतरनाक है। इसलिए मैं कहता हूँ कर्मणि कुशलो यस्तु, जो काम करने में कुशल है, श्रद्धावान से समर्पित, श्रद्धावान होकर के और समर्पित होकर के जो काम करने में कुशल है, शास्त्र कहता है इच्छा तस्य सर्वत्र, उसकी डिमांड सारी दुनिया में है। जो स्किल्ड है, जो काम करने में प्रवृत्त करता है, उसकी डिमांड हर एक कोई करता है, भाई मुझे तो वो आदमी दे दो। हाँ भी कई बार रिश्ता साल के अंदर सर ये इसको नहीं तो और कहीं विभाग में भेज दो, क्योंकि ये ना काम करता ना करने देता है, ये इनको तंग करते हैं। तो फिर कई बार क्या होता है कि वो लोग वो फिर कहते हैं किसको भेजा, साहब उसको भेजो वहां पर काम कर रहा है। भाई क्यों, साहब बहुत बढ़िया काम कर रहा है। ठीक है, कर्मणि कुशलो यस्तु श्रद्धावान से समर्पित, अपेक्षा तस्य सर्वत्र, उसकी जो डिमांड है, उसकी मांग है सारी दुनिया में है, देश में है और विदेश में है। और साधारण संस्कृति व्यक्ति सभी के द्वारा आदर पाता है। बताइए, शास्त्र की बात मानोगे या अपनी वो चीज मानोगे जिसमें नेगेटिविटी फैलाना चाहते हो इस संस्था में और समाज में, देश में। से व्यक्तियों की आवश्यकता नहीं है, में आवश्यकता है जो जज्बा रखता है। राजपूत जैसा व्यक्ति आते ही एकदम उछाल आ गया, उसके नेतृत्व में आप सबने काम करना शुरू किया, मुझे आप पर सब पर बेहद खुशी होती है। मैं आपको भी सब में साथ ले रहा हूँ। सब तामझाम कुछ नहीं था, कुछ नहीं था तामझाम। दिखाई देता हमारा कुलपति के पीछे गंदी का ढेर था, आज वहां पर सब क्या है, नर्सरी बनाई, किसने बनाई, लगे फार्मिंग कंपोस्ट है, उधर शुरू हो गया। छोटी चीज है, छोटी चीज बड़ा काम करके जब बनता है यार, छोटा छोटा काम करके बड़ा महान बन जाता है। तो आप सब लोगों से मेरा यही संदेश है कि हम को उस दिशा में ले चलें, जो मंतव्य, जो ध्येय, जो इच्छा उस बाबा की थी जिसने अपना सर्वस्व में उछाल कर दिया, जो गोली खा गया सीने पर। आप उस संस्था में काम करते हैं, जिस बाबा ने गोली खाई, का स्टैचू चांदनी चौक के घंटाघर पर इतना बड़ा शानदार, बाबा का बंगलौर में उसके नाम से रोड है, श्रद्धान मार्ग। आप कीमत समझते हैं उसकी? नहीं समझते ना, यहाँ से बाहर निकल के नहीं समझते, यहाँ भी नहीं समझते हैं। अपनी पहचान यहाँ बनाइए और विदेश में बन। आज इनको विदेश में भी जानते हैं, दस्ता साहब को, दुबे जी को भी जानते हैं। तमिलनाडु मेरी बेटी पढ़ती थी, वहाँ किताब, इनकी किताब पढ़ रही थी, तो उसमें लोगों ने देखा ये तो हरिद्वार के हैं, बोले ये मेरे मेरे पापा के कुलीग हैं। ये काम जा रहा है ना, चलेगा। आप आगे बढ़ें, बढ़ेगी, और जितना आप काम करेंगे उतना ग्राफ आपका ऊपर उठेगा। नहीं करेंगे नीचे जाएगा, निश्चित रूप से जाएगा। मैं यज्ञ वेदी पे कह रहा हूँ कि आप लोगों को कड़वी से कहता हूँ, आपका कोई अहित नहीं होगा यहाँ पर। थोड़ा कहीं हो गया होगा, अहित आपका नहीं होगा कि नौकरी नहीं जा रही है, गलती होगी उसकी गलती से होगा जो होगा, लेकिन प्रशासनिक रूप से कुछ नहीं होगा। शांति से अपने जज्बा के साथ और अपने पूरे उत्साह के साथ काम करें, मैं वो दिन देखना चाहता हूँ।
सभी लोगों का गुरुकुल कांगड़ी के इस वर्चुअल प्लेटफॉर्म पर स्वागत है। आप सभी से निवेदन है, थोड़ी देर का इंतजार करें। सेशन हम कंटिन्यू रखेंगे, लेकिन उसमें थोड़ा सा, तो आप सभी से निवेदन है, कृपया जुड़े रहें।
इसमें प्रैक्टिस सही जाना बोले तो हम लोग मानसिक के साथ उसमें बताया क्लास में चल हाँ कुछ हाँ पहले चार पाँच जैसे देखते वैसे इसके को लगाओ तुम मार्कशीट का और उसकी एक बनाओ पीडीएफ है ना दोनों की अलग अलग पीडीएफ बनाओ चारों की हाँ उससे ले लो फोटो कॉपी ले लो
So good morning. Hello. Good morning, everyone. Myself Vinod Nod, behalf of Department of Pharmaceutics. I welcome you all on today at Padap MahaKumbh. Are going today World Science Day.
मैं से वो लेकर आया हूँ
Lot of participants
चने और पराठे
joined. At first, I welcome you all, sir. First of all, I want to invite Dr. Deepika Singh. Ma'am, are you there? No? Dr. Sanjay Singh, sir, are you there? Hello, Dr. Sanjay Singh.
Yes, sir. Good morning. Good morning, sir.
Good
You are getting my voice?
Yeah, I'm getting your voice.
Okay, sir. Thank you so much. Good morning, all of you.
And sir, I request you to introduce.
I am audible to you.
Yes, sir.
Sir, I request you to
When will this function start?
today in the clear mind.
Okay, sir. Thank you so much, sir.
Go ahead.
Honorable Professor R.K. Shastri, sir, Vice Chancellor of Bakul Kangri University. Our chief guest, Professor Sunil Joshi, sir, Vice Chancellor of Uttarakhand Ayurvedic University, Dehradun. Guest of honor, Professor Ranjit, sir, Vice Chancellor, Sobit University, Gangu. Our respected speaker, Professor Anurag Verma, sir, Director, Tirthankar Mahavir University, Muradabad. Organizing Secretary, Dr. Bipin Sharma, sir, Dr. Deepika, Dr. Vinod Notial, and other dignitaries and all the participants. A very good morning to all of you. It's my privilege today. I am going to introduce our respected speaker, Professor Anurag, sir, in a national seminar on the occasion of the National Science Day. The Dr. Anurag has a huge academic profile. So today is not enough time for us to tell his profile completely. And I am going to give a short introduction of Dr. Anurag. Sir is a professor in pharmaceutical science and principal at College of Pharmacy, Tirthankar Mahavir University, Muradabad, Uttar Pradesh. Sir has completed his M.Pharm in pharmaceutics from KLE College of Pharmacy in 1999 and PhD in the pharmaceutical sciences from Uttar Pradesh Technical University in 2012. Sir has a huge experience about the 21-year teaching and research experience, and sir also has a 2.5 year of industrial experience. He has published more than 70 research and review articles in the reputed national and international journals. And sir has 2 book chapters to his credit in Spiritual Nature and elsewhere. This is a short introduction of Dr. Anurag. Now I am requesting to Dr. Vinod Notial, sir, please take the session ahead. And thank you so much and welcome. I am welcoming to our respected speaker, Dr. Anurag Verma, sir, Sunil Sharma, sir, and all the dignitaries those have participated and joined in this session. Thank you so much. Thank you so much, all of you.
Yes, sir, you introduce the speakers now.
Thank you, Sunil.
Radio time.
I am audible.
Yeah.
Now I would like to request Dr. Sandeep Bedalanka. संदीप जी आप हैं क्या?
हाँ
संदीप जी अब बात करने से पहले अब अनुराग सर के लेक्चर से पहले हम आपसे रिक्वेस्ट करना चाह रहे हैं कि आप वेद मंत्रों से इसका प्रारंभ करें.
I think you have asked me to say something.
डॉक्टर संदीप बेदाला.
सर आपके पास हेडफोन है क्या?
Yeah, headphone is there.
हेलो डॉक्टर संदीप जी.
Doctor, I think he might be.
सर जी.
संदीप बेदालंकार जी आप सुन पा रहे हैं?
जी.
संदीप जी आप शुरू करें प्लीज. जी
प्रारंभ करो.
वेद मंत्रों की शुरुआत करें.
अथ यजुर्वेद 22वें अध्याय का 22वां मंत्र है. ओ
आराशे राजा ईशव्यो मार्गो जायता धोद्रिधेनुर्मूढा नासो शक्ति योगा जिष्णुरथेष्टा सभयो योगाश्च यजमानस्य जायता निकामे निकामे नः पर्जन्यो वर्षतु फलवत्यो नयोषधया योगक्षेमो नः कल्पताम् 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 कल्पताम्।
आपने वर्षों से वचन आपका बहुत पहले भी कर बहुत बहुत धन्यवाद संदीप जी, और हमें उम्मीद आगे भी जो है का करते रहेंगे। बहुत बहुत धन्यवाद। इस क्रम में हम आगे अपना लेक्चर
आपकी आवाज बहुत तुम ध्यान दो।
माइक्रोफोन
आवाज बहुत कम आ रही है, आवाज का पता नहीं चल रहा।
Hello.
Hello.
Am I audible now?
No, yeah, it's okay.
Hello. I'm
Now
audible
it's,
now.
yeah, now it's okay.
Okay. Okay. Thank you, sir. Thank you.
Yeah.
On continuation, now I would like to request
सर आप
Professor Anurag
लेकर
Verma
आए हो?
to deliver his lecture. Professor Anurag Verma, sir, am I audible to you, sir?
Yes, you are
सर
audible
किसी
to
से
me.
पूछना, किसी के
Yes,
पास
sir.
earphone
Please
है
go
क्या,
ahead, sir.
कोई भी कैसा भी ईयरफोन हो।
Okay. Thank you very much for. I'm grateful to the organizing committee for giving me the opportunity, and I'm also thankful to the Vice Chancellor, GKV.
ईयरफोन
गुरुकुल
मतलब वो कौन से वाले कान में डालने वाले हैं? अब
मुझे इस लायक उन्होंने समझा कि आपके विश्वविद्यालय में नेशनल साइंस डे के उपलक्ष्य में कुछ अपना वर्क प्रस्तुत करूं। So today's the topic of my talk is role of nanotechnology in the treatment of visceral leishmania. Am I audible? Am I audible?
Yes, sir. Sir, good. Anurag sir, okay.
Shall I proceed? Okay.
Yeah.
Okay. So I will start with what is leishmaniasis. It is a parasitic disease that is found in the parts of tropics, subtropics, and southern Europe. It is caused by infection with intercellular protozoa parasite of genus Leishmania, which are spread by the bite of infected sandfly. There are other modes of transmission, which include conjugal transmission, contaminated needle sticks, transmission, sexual intercourse, etc. Leishmaniasis affects some of the poorest people on earth. That is with malnutrition, population displacement, poor nutrition, weak immune system, and lack of financial resources. Basically, it is a disease of poor. Now, types of leishmaniasis. There are three main forms of the disease: visceral leishmaniasis, cutaneous leishmaniasis, and mucocutaneous leishmaniasis. Visceral leishmaniasis in our countries is known as Kalajar, and cutaneous leishmaniasis is the most common form of leishmaniasis. India is endemic for both visceral leishmaniasis and cutaneous leishmaniasis. In our country, there are around 130 million people at risk of Kalajar in 54 districts of 4 endemic states: Bihar, Jharkhand, West Bengal, and Uttar Pradesh. Mr. Vipin, I am getting disturbed.
मैं जा रहा हूँ तो सर उससे बतिया के आते हैं.
Hello,
क्या नाम है उसका?
Mr. Vipin. Hello.
आकुल से.
Yes, sir. Yes, sir. You are audible.
You are audible.
I am getting
से बस वो लेकर
I'm getting disturbed.
एक सर की वो हो रही है.
Okay. So, visceral leishmaniasis is a serious, progressive, and potentially lethal systemic disease. It is characterized by irregular bouts of fever, weight loss, enlargement of the spleen, liver, and anemia, and it remains one of the top parasitic diseases with outbreak and mortality potential. Most cases occur in Brazil, East Africa, and eastern part of in our country. Cutaneous leishmaniasis causes skin lesions, mainly ulcers on exposed part of the body, in lifelong scars and severe disability or stigma. About 95% of the cutaneous leishmaniasis cases occurs in Afghanistan, Algeria, Bolivia, Brazil, Colombia, Iran, Iraq, Pakistan, and the Syrian Arab Republic, Tunisia, and recently it has also been reported in Rajasthan, in our country. Localized cutaneous leishmaniasis often spontaneously resolve in 3 to 6 months without therapy, although some infections persist indefinitely. Mucocutaneous leishmaniasis leads to partial or total destruction of mucous membranes of the nose, mouth, and the throat. Over 90% of the mucocutaneous leishmaniasis occurs in Bolivia, Brazil, Ethiopia, and Peru. Now, the pathogenesis of leishmaniasis. It is caused by the bite of sandfly. The bite of one sandfly, infected sandfly, is sufficient to cause the disease, because a sandfly can ingest more than 1,000 parasites per bite. Only the female sandfly succeeds the protozoa, infecting itself with leishmania parasites contained in blood. It sucks from its human or mammalian host.
Excuse me, Anurag sir. Anurag sir, sorry to intervene you.
Hello, सुन रहा हूँ मैं.
Anurag sir, your presentation is not visible, sir.
Not visible.
Sir, your presentation is not visible, sir. We are not seeing your screen.
Yes, please.
Sir, your screen is not being shared, sir.
Now? Is it shared?
Yes, sir. Now you are starting. Yes, sir.
Shall
Yes,
I
sir. Now
once
your
again?
screen is being shared.
Shall I start once again?
Okay, thank you, sir.
No, sir. Anurag sir, from where itself? Because otherwise they are.
Okay.
Yeah,
so
let's
now,
start from where.
once bitten by the sandfly, over 4 to 5 days, the parasite continues development inside the sandfly, and where it undergoes a major transformation into the promastigotes form. As an infected sandfly bites a mammal, the promastigotes are infected under the skin, and these promastigotes are infected by the macrophages, and parasitophorous vacuoles are formed. In macrophages, the promastigotes are converted into amastigotes, and this amastigate undergoes binary fission, and large number of amastigates form and lead to death of macrophages, which leads to lasting and infection of the macrophages. This transition of promastigate to amastigate is must for the transmission of disease, as amastigates are expert at exploiting host cell process and establish infection and persist in the system. A large number of replacement of macrophages lead to enlargement of the visceral organs, like visceral means internal organs like liver, spleen, heart, abdomen, and which are linked with the reticular endothelial system. That decrease in red and white cells lead to anemia, leukopenia, respectively, secondary bacterial infection, and fatigue. The species of the parasite and the host status. Parasites may incubate for weeks and months before presenting the disease. Pharmacotherapy of leishmaniasis. The treatment mainstays of leishmaniasis are the pentavalent antibody compounds first introduced in 1938, for example, sodium stibogluconate, alumine, but unfortunately resistance to this agent is on the rise. In Bihar province of India, where visceral leishmaniasis is endemic, resistance is as high as 43%. Amphotericin B is effective against pentavalent antibody resistant mucocutaneous disease and visceral leishmaniasis. Its use is limited because of its toxic adverse effect profile. Liposomal amphotericin B is more active, better tolerated, and are being used as visceral leishmaniasis, but its treatment is costly. Meltefosine is the sole oral agent that has been shown to be effective against leishmaniasis.
Make sure your device is connected correctly and right.
Intramuscular pentamidine is effective against visceral leishmaniasis, but this drug is associated with persistent diabetes mellitus, and disease recurrence. Orally administered ketoconazole, itraconazole, fluconazole, allopurinol, and dapsone have been examined internationally, but none is as effective as pentavalent antibody compound. Topical paramoxicillin have also shown to be effective against cutaneous leishmaniasis caused by L. major and L. mexicana. Now, why do we need nanotechnology to treat leishmaniasis? Please wait. Okay. The reasons are the conventional therapeutic interventions are not effective as leishmania parasites hide in long-lived macrophage cells surviving hostile conditions. Nanotechnology-based immune systems can penetrate the macrophage cells and infectious parasites, enabling targeted and efficient delivery. Nanotechnology-based drug delivery system enhances the efficacy by regulating the pharmacokinetics and reduce drug toxicity with sustained release of drugs. Various nanotherapeutics have been approved by the Food and Drug Administration and are currently available for clinical use. Please, I am getting
Yeah. It's my humble request to organizers. Please unmute all the participants who are there attending this lecture, because lot of
very sorry for the disturbance. I got a call from the Okay. Now, why solid lipid nanoparticles to treat leishmaniasis? Solid lipid nanoparticles have gained importance. The matrix of the lipid core has significant role in regulating the release pattern and protect the loaded drug from chemical and
माइक्रोफोन है सर के लैपटॉप में सर के अब सिस्टम लगा हुआ है.
Hello, I am audible. Hello. 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 Am I audible? Am I audible? Hello. Hello. Am I audible? Am I audible? Hello. Hello. Am I audible? Okay. So endogenously, the SLNs are conducive for enhancing the oral uptake of therapeutics by retaining a solubilized state of drug in the GIT and favor the formation of mixed micelle by inducing the secretion of bile salts and phospholipids. Now, innovative solid lipid nanoparticle approaches. So recently we have worked on the development of novel drug release system for the treatment of leishmaniasis utilizing the solid lipid nanoparticle approach. So we have developed two types of nanoparticles. One is modified solid lipid nanoparticles, which uh, uh, contained two drugs, amphotericin B and paromycin, because monotherapy often fails to treat leishmaniasis. So we have used two drugs, amphotericin B and paromycin. And these solid lipid nanoparticles are made up of glycerol monostearate coated with 2-hydroxypropyl beta-cyclodextrin. And the second approach is modified solid lipid nanoparticles bearing amphotericin B coated with vitamin B12 stearic acid conjugate. Actually, this is the work of senior research fellow Shabi Parvez, was working in INST, Institute of Nanosciences and Technology, Mohali, and the work was carried out under the joint guidance of me, Dr. Shyam Sundaram from BHU IT, and Dr. Shyamlal from INST, Scientist D, INST. And this work was published in a scientific report with the topic is modified solid lipid nanoparticles encapsulated with amphotericin B and paromycin, an effective oral combination against experimental urine vessel leishmaniasis. It should be noted that both the drugs amphotericin B and paromycin are generally used by parenteral mode, but here we have developed them into an orally administered dosage form. So in this approach, we exemplify the preparation and characterization of HPCD modified solid lipid nanoparticles based combinational cargo system of amphotericin B and paromycin. As I already told that monotherapy with anti-leishmanials has led to the development of drug-resistant parasites. Therefore, multi-drug combination strategies have emerged as a viable option. Recent findings have shown a synergistic interaction between amphotericin B and paromycin in experimental and clinical visceral leishmaniasis. Co-treatment with paromycin and amphotericin B has shown synergistic effect in leishmania-infected mouse peritoneal macrophages. Now, these modified solid lipid nanoparticles were prepared by emulsion solvent evaporation approach, and then these were coated with 2-HPCD using trehalose as a cryoprotectant. Why we have used HPCD for the coating is that the HPCD has the potential to improve the macrophage specificity by membrane fusion, vesicular transport, or cytoskeletal remodeling. And because of this, by coating the solid lipid nanoparticles with HPCD, resulted in increased uptake of these nanoparticles via the by macrophages. More HPCD being it is not digested in the stomach. So because of this, it also prevented the lipolysis of the solid lipid nanoparticles by the gastric lipases. And it also prevents the faster drug release due to lipid erosion under the effect of fluid and enzymes present in the intestinal tract. The size was found to be of the plain blank nanoparticles, it was 38 nanometer. Drug loaded, it was 141 nanometer, and coated nanoparticle, it was 64 nanometer. And there was very good entrapment efficiency of amphotericin B being lipophilic in nature. These are the SAMs and came of these our solid lipid nanoparticles. The solid lipid nanoparticles were characterized for various parameters, say stability and in vitro drug release. In vitro drug release were carried out in acidic pH 1.6 and alkaline pH conditions to determine the stability of SLN and while they transit through gastrointestinal system for stable administration of these nanoparticles orally. The amphotericin we release from the lyophilized uh, modified solid lipid nanoparticles, which are known as MDDSLN, was found to be around 0.33. There was only 0.33% released in the stomach for up to 2 hours, and in SGF, and only 4.2% of drug released in up to 4 hours in stimulated intestinal fluid. While for PM it was 10% in 2 hours in SGF, that is stimulated gastric fluid, and 14% for 6 hours in stimulated intestinal fluid. It was assumed that the drugs present on the surface of the nanoparticle, which is closely bound, released in the stimulated gastric fluid. Up to 57% amphotericin B paramycin was released in 24 hours. We have also carried out the cytotoxicity study on J774 A.1 macrophage cell line, and it was observed that the drug alone, that is amphotericin B and paramycin, causes more cytotoxicity compared to cytotoxicity caused by the HPCD coated solid lipid nanoparticles. Macrophage, we have also carried out the macrophage uptake study, and it was observed that the HPCD modification enhanced the uptake of solid lipid nanoparticle by macrophages, where Leishmania amistrigotes dwell amply. And it is the diagrams which shows the we have used a number of dyes to show that you see that the nanoparticles they are rapidly uptake by the macrophage. In vivo toxicity studies we have also carried out on the same cell line. In vivo anti-leishmanian activity of modified. Am I audible? Hello? Am I audible? Hello? 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 Am I audible? In vivo anti-leishmanian activity we have also carried out using the L. donoveri infected Balpsky mice, and it was observed that these nanoparticles exhibited significant anti-leishmanian activity against L. donoveri infected mice model upon oral administration. So in this study, modified solid lipid nanoparticles of amphotericin B and paramycin was successfully prepared by emulsion solvent evaporation method by using HPCD as a surface modifier for the enhancement of oral viability in the treatment of DL. The prepared modified solid lipid nanoparticles showed enhanced efficacy in comparison to the liposomal AMB in both in vitro and in vivo by reducing the intracellular amastigates. Hello. Intracellular amastigate growth in L. donoveri infected macrophages and hepatic parasites burden in L. donoveri infected Balpsky mice. The ability of such nanoparticles support promising utility as a drug delivery system as an alternative to the conventional therapeutic towards the infectious disease. Is there any question? Is there any question? Hello? Hello, is there any question? Is there any question? Okay. So I will start the new approach. Modified in the next approach, we have prepared the modified solid lipid nanoparticles bearing amphotericin B coated with vitamin B12 stearic acid conjugate, and this is the work of working in INST, and the work was carried out in the joint supervision of me, Dr. Shyam Sundar, and Dr. Shyam Lal, and it was formulation, characterization, and in vitro anti-leishmanian evaluation of amphotericin B loaded solid lipid nanoparticles coated with vitamin B12 stearic acid conjugate, and was published in Material Science and Engineering C. Here we have formulated a novel nanoparticulate system of vitamin B12 stearic acid conjugate coated with solid lipid nanoparticles encapsulated with amphotericin B to improve amphotericin B oral absorption and enhance the anti-leishmanian activity and safety. To the best of our knowledge, no study reported by using B12 stearic acid conjugate coated with SLN as a carrier for amphotericin B as a potential therapeutic strategy against visceral leishmaniasis. Am I audible? Hello, am I audible?
Sir, you are audible, sir. Please go ahead,
Okay.
sir.
So these nanoparticles are prepared by using a little bit of chemistry. There is one is Oxima method, which is generally used for, which we have used for the preparation of vitamin B12 stearate. And here, B12 stearate was prepared by in the presence of a carboxyl activating agent and oxima in the presence of 6 millimole sodium bicarbonate. Mixture was continued agitated for 12 hours at room temperature. After agitation, vitamin B12 stearate was separated. Now, vitamin B12 stearate modified solid lipid nanoparticle. Vitamin B12 was reported to enhance the rate of phagocytosis. The presence of vitamin B12 on the surface of solid lipid nanoparticles lead to enhanced uptake. Vitamin B12 coated vitamin B12 stearate nanoparticle to receptor-mediated endocytosis. Amphotericin B has been reported to exhibit higher partition coefficient when stearic acid was selected for preparation of solid lipid nanoparticles. Also, saturated fatty acids with high melting point showed high permeation rate across the biological membrane, and vitamin B12 as an effective targeting agent for nanoparticles, which resulted in enhanced uptake of nanoparticles from the intestinal lipids. Particles. Plain nanoparticles and double nanoparticles are found to be 101 and 303.66 nanometers. Intraprinter efficiency was found to be higher, and it is around 97%. In vitro release was carried out in the media 7.4, and in vitro cytotoxicity study was also carried out on by MTTSA on J774A cell line. Anti-leishmanial activity of these vitamin B12 stearate coated amphotericin B loaded solid nanoparticles was also tested on aldonomin infected J774A1 macrophage cell line. So in the current study, a novel nanoparticulate drug delivery system was developed for the increased oral delivery of amphotericin B. The particle size distribution indicated homogeneity of synthesized solid lipid nanoparticles with a net neutral charge on these nanoparticles, favoring higher mucus permeating ability. Oral bioavailability of amphotericin B. Need vitamin B12 stearate coated amphotericin B loaded solid lipid nanoparticles confer complete internalization within the macrophage cell. In vitro studies showed lower cytotoxicity against J774A1 cells and higher chemical stability against digestive enzymes. Thank you. Now, if you have any question, please may ask.
सर ने क्वेश्चन आंसर सेशन के लिए बोल दिया।
किसी का क्वेश्चन है तो
अगर आप कोई क्वेश्चन पूछना चाहते हैं तो आप पूछ सकते हैं।
Hello. I
Hello.
would like to request all the participants, if you have any queries, any questions, please you can write in chat box or you can directly ask.
डिफरेंस बिटवीन एसएलएन बोलो
मैं बोलूं
सर इज द क्वेश्चन
sir, there is a question in chat box and question is, please tell difference between SL and metal nanoparticles. Sir.
SLN is solid lipid nanoparticles, right? We are using a lipid, right? And why we are using lipid? Because lipid, all our biological membranes are made up of lipid. So it is assumed that there will be better say partition of these nanoparticles through our biological membrane. Hello, any other?
Any other question, please? Sir, I think there is no more question. And now I would like to request Dr. Deepika Saini. Ma'am, am I audible to you, ma'am? Are you there?
Yes, I am audible.
Okay.
Hello.
And before going to the finalization of this session, now I would like to request Professor Sunil Sharma, sir.
Yeah, I'm here, dear. Hello, am I audible?
Yes, I am audible.
Yeah. So, first of all, I would like to tell that Anurag Verma has delivered his talk in a very nice way, and what I feel that all the participants would be benefited out with his presentation. The presentation is quite simple and quite nice, and things were delivered by him. Again, a great effort made by Dr. Professor Anurag Vermaji. So I extend thanks on behalf of organizer, and not only that, I also extend my thanks for inviting me and asking me to chair a session in which number of participants has joined, and they all were benefited out of this.
concluding
Thank you, sir. Thank you very much, sir.
Okay.
And finally, I would like to request Dr. Deepika Saini for presentation of the vote of thanks. Ma'am Deepika, ma'am.
Yes, sir, I'm here. First of all,
Hello.
good afternoon, everybody. Hello. Yes, sir, am I audible?
Yeah,
Hello?
whatever, ma'am. Yeah,
Yes, ma'am. Please go ahead, ma'am.
All right. Yeah, good afternoon, everybody. This is Dr. Deepika Saini. I'm working as an assistant professor, Department of Zoology, Chamanal Mahavidyalaya, Landora, Haridwar. First of all, I would like to thank all the organizers of today's seminar. As we know, we are here using online system. Exactly why, as we all are in a pandemic situation right now, and it's a kind of exactly, it's not a lockdown, but a kind of. So we are using this online portal. So on the occasion of today's national seminar on National Science Day 2021, having an outstanding topic, kind of subtopic, that is future of STI, impact on education, skill, and work. So I'm here, all of you, to propose a vote of thanks to all the active participants over here. First of all, I would like to thank our Honorable Chancellor, Dr. Satyapal Singh Ji, our Honorable Vice Chancellor, Professor Rup Kishore Shastri Ji, Dean of Faculty of Medicinal Science and Health, Professor R.C. Dubey Ji, Head, Department of Pharmaceutical Sciences, Professor Satyendra Kumar Rajput Ji, our Organizing Secretary, Dr. Vipin Sharma Ji, our Convener, Professor Satyendra Rajput Ji, our esteemed Chief Guest, Professor Sunil Joshi Ji, and Guest of Honor, Professor Ranjit Singh Ji. Thank you so much of all of you for giving your precious time in the today's inaugural session. I would like to propose a special vote of thanks to Professor Anurag Verma Ji, as his PPT presentation on Leishmania was an outstanding one. We got to know so many things out of it, and I hope all the participants out here, they definitely have benefited themselves. So without wasting any time, I would like to thank once again all of you for your participation and making today's inaugural session thank you so much. I would like to hand over for the further proceedings to Dr. Vipin Sharma, please. Thank you so much, guys.
Hello. Thank you, everyone, and especially thanks to the Anurag Verma sir and to the chairman, Professor Sunil Sharma sir, Deepika ma'am, and all the participants and who joined us through that online and offline also. Thank you very much and have a good day, sir.
Hello. Am I audible, sir?
Vipin sir, you are audible.
Sir, thank you, sir. Thank you, all the dignitaries, all the participants for their presence here. Professor Anurag Verma, sir, from MahaKumbh University, our chairman, sir, Professor Sunil Sharma, sir, and our moderator, Dr. Deepika Seni ma'am, our coordinator, Sanjay Singh from Institute of Pharmacy, Dehradun, and all the persons associated directly, indirectly, offline and online mode for being present in this session. So once again, thank you, all of you, sir. And now I would like to invite our colleague, Dr. Kapil Goyal, sir, for coordinating the next session. And next session is being chaired by Dr. V.K. Bastia, sir, from AIPS Rishikesh. So once again, thank you, all of you, sir. Be present with us.
Thank you, Vipin. Thank you very much for inviting us.
Namaskar, sir.
Namaskar.
For your blessings. And I would like to invite you, sir. Please come to Haridwar.
Yeah, definitely.
And visit our department, sir.
Yeah, definitely. My pleasure to be there. Okay, thank you once again, Anurag sir, for nice presentation.
Now it's over to you, Dr. Kapil Goyal, sir.
Thank you, sir. Very good afternoon to all of you. This is Kapil Goyal from Guru Nanak University. I welcome you here. Dr. Anurag sir is, I think, available there. Now, I welcome to Dr. Binay Kumar Basitia from the AIMS Rishikesh. Sir, welcome in Guru Nanak University, Haridwar. And Dr. Basitia is a professor in the Department of Forensic Science and Toxicology in the AIMS Rishikesh. So I welcome you, sir, on the behalf of Guru Nanak University and organizing team of the Aushadhi Padak MahaKumbh and the National Science Day organizing committee. All are welcoming you in Guru Nanak University, Haridwar.
Thank you.
Dr. Prashant Gathori is also with us. I welcome you, Prashant Gathori, sir. He is a coordinator of today's session, and I welcome you here. Prashant sir, your mic is, I think, paused. I also welcome to Bodke sir. Bodke sir. Surinder H. Vodke sir is working in the Guru Ghasidas Vishwavidyalaya, Bilaspur. He is the person who had taken my examination during my B.Pharm. So I think sir is remembering me or not. I was the student in the Sudha Bhagwan Singh that time.
Yeah. I remember, sir.
So sir, welcome in Gurukul Kangdi University, Haridwar.
Thank you.
Dr. Surendra is working in Guru Ghasidas Vishwavidyalaya, Bilaspur. He has published a lot of research paper related to his field, and 6 students are awarded PhD under the supervision of the Professor Vodke. I welcome you for this session. Sir, Dr. Gatori will coordinate the session. So, Gatori sir, you can please continue. I also welcome Dr. Professor Nitin Sharma. Nitin Sharma is there? I think he's not joined. Professor Nitin Sharma from MIT, Meerut. He is also the moderator of this session. So I welcome you, sir, in Gurukul Kangdi University, Haridwar. So on the behalf of the National Science Day Organizing Committee, I hand over the mic to coordinator of this session, Dr. Prashant Gatori. He is working as a associate professor in Graphic Era Hill University, Dehradun. He is a very good researcher and completed his projects in the IIT Roorkee also. And personally, I know him since 2000. I think 2000, Prashant. We were.
Yes, sir. Yes, sir. Yes, sir.
We are the alma mater of the same institution. So I welcome you here. Sir, please continue the session.
Okay. Am I audible? Please check.
Yes, audible, sir. Audible.
Okay. It's been a great pleasure to interact with talented pool of pharmacy fraternity. So it's always been a great pleasure. So Surendra Vodke sir needs no more formal introduction. He is a well-known personality in the field of pharmaceuticals. So I remember the day when we were referring to the GPAC, and we were fortunate enough to find a book written by Vodke sir. So without taking much of time, I'm just going to invite Dr. Surendra Vodke sir to start his presentation.
Thank you, Prashant. Thank you, Prashant. First of all, I would like to thank the organizing committee for giving me an opportunity to have interaction with all of you. Am I audible?
Yes, sir, you are audible.
So I'll start my presentation.
Yes, sir. Yes, sir. Sir, your screen is visible, sir. Please continue, sir.
Okay, yeah.
Thank you, sir.
Today's topic of my presentation is the use of experimental animals in research with special reference to CPCC and other guidelines. Why I have chosen this topic because nowadays, because of this COVID-19 situation, much emphasis is being given on the experimental animals for research. Our aim should be to take care of these animals, and this is our this must be done with the guidelines which are given by the government of India. COVID-19 worldwide health emergency situation or the pandemic caused by this. caused by this COVID-19 has caused the confinement of people to their homes. And since the outbreak, more than 1 million people have lost their lives. The global economy is expected to contract tremendously. The millions of jobs have already been lost, and there appears to be no end to COVID-19 situation yet. In many countries, the number of new COVID-19 cases are rising at alarming rate, and the second wave is already an unwelcome reality. This is the situation right now, and much uncertainty remains about how and what will happen, and it has seriously set back development progress around the world. So many countries had started re-examining their supply chain of critical care, health and livelihood related products. Has led to a surge of nationalism. I give stress on this word because nowadays people are thinking about their own products, and they have started working on this line, and they have started on much emphasis is being on the research, and particularly related to the pharmaceuticals, medical supplies and equipments which are used domestically should be manufactured, or domestically research should be done on this in this area. So even countries that traditionally had no capability in these areas have initiated to develop the same. India had already had the capability, and they are trying to strengthen this in future, and they have started working on in this area. This one statement, that is the one good thing caused by COVID-19. Many people will not agree with me because this is not a good thing, but one good thing that is caused by this COVID-19 is the innovation. And this was a statement given by Honglio Albert Galasso of the Harvard Business School on 7th May 2020. And this is the reality. This is the fact that people are now thinking about the research. Otherwise, even the government was not taking much care of this area. Nowadays, there are various agencies, or there are various institutions, research institutions or universities. They have started or giving emphasis on research. One of them is the Tata Consultancy Services. They have started giving much, they are giving a lot of work is being done by these Tata Consultancy Services. Even Indian government has poured crores of rupees in research. Government of India is aiming to increase the healthcare spending to 3% of the gross domestic product, that is GDP, by 2020, and this must be welcomed by all of us. India ranks 3rd worldwide for pharmaceutical production by volume and 14th by value. The country has estimated domestic pharmaceutical industry with a strong network of 3,000 drug companies, and there are about 10,500 manufacturing units throughout the country. Now, what is the importance of animals in research? Because these animals which we use, they are important. Animal experimentation has contributed to an increase in average life expectancy of about 25 years since 1900. 19th century. If you start with the 19th century, about 25 years age has been increased, and this is only because of the experiments on animals. This is the great contribution of these experimental animals in our life. Research involving laboratory animals is necessary to ensure and enhance human and animal health and protection of the environment. In the absence of human data, research with experimental animal is the most reliable means of detecting important toxic properties of the chemical substances and for estimating risk to human and environmental health. This is how the animals are important for us, because we cannot directly work on the humans, and therefore research should be carried out in the animals, and this is one of the most reliable source of information of data. Research animal must be used in a responsible manner. This is the important thing that should be taken care of by everyone. The animals should be used in a responsible manner. Scientifically, valid research are designed to reduce, refine, or replace the need of the laboratory animals and this is encouraged. And this is called as the 3R principle: reduce, refine, and replace. Nowadays, 4th R principle is added, that is called as the reuse of the animals. Wherever possible, the animals should be reused, and this should be with the proper permission from CPCC or IAEC, whatever is applicable. Now, why is there a need to use animals? When any chemical or any drug which is introduced in our body or in the living animal, it is converted into another form, and this form may be beneficial or it may be toxic. What we call as the metabolism, and this cannot be recreated in any other situation. For this purpose, the animals are required, or the living body is required, and the most reliable source is the animal, experimental animal, and therefore there is a need to use this animal. So using animal in experiments is critical because such complexity cannot be duplicated in cell culture or in non-living system, which I have already explained. Scientists use whole animals to understand the relationship between the exposure of the drugs and their effects on human body. And what is the responsibility of the researcher or scientist? The responsibility is to take proper care of the animals, to use them appropriately, responsibly, and humanly. Responsibly and humanly. Appropriate means the number required. That should be appropriate. Unnecessary number of animals. If you are increasing the number of animals, if suppose 6 animals are required and you are taking 10 animals or 15 animals, no, not at all. You should use minimum number of animals wherever possible, and you should take the proper care of these animals. After the experiments are over, you should take their post-experimental care, and they should be treated humanly. One should take care of these animals. Data from these studies provide the initial foundation to distinguish and recommend which level of exposure is safe and which is harmful. This can be done only in the animals. If you see the use of these animals, how they have contributed in our life, These are the major achievements in basic research that depended on animal experiment. If you see the 16th century, discovery of blood circulation, which was done in animals. The 17th century, the measurement of blood pressure, that is now possible because of the studies on animals. 18th century, vaccination to stimulate immunity and understanding of infectious diseases. Nowadays, this immunity is important in case of COVID situation. And nowadays, vaccines are developed, have been developed, and we are using these vaccines in our country and worldwide. This is because of the use of animals only. 19th century, discovery of and understanding of hormone systems. This was also possible because of the animals. If you see the 1920s, the discovery of vitamins. 30s, discovery of mechanism of nerve impulses. Discovery of tumor viruses. 1940s, understanding of development. 1950s, understanding of control of muscle activity, and so on. Discovery of monoclonal antibodies and their function in the liver. 1970s, understanding of transplantation, antigens, understanding the way of the brain functions. Discovery of prostaglandins, major breakthrough. 80s, development of transgenic animals, understanding of the basics of memory. This was because of the animals only. 90s, autoimmune disorders, and so on. Even 70s, drugs were discovered to treat the ulcers. For the treatment of asthma, drugs were discovered during this period. For leukemia also. In 80s, immunosuppressants, scanning of for improved diagnosis. These were because of the animals. 1990s, laparoscopic surgical techniques, breast cancer links, gene therapy, etc. All these things were possible because of the animals. So how exactly has animal research helped us and our family? Polio, diphtheria, mumps, measles, rubella, and so on. This is because of the animals. Severe burns, juvenile diabetes, leukemia, newborn sickness, premature births, etc. Prevention and treatment of birth defects. Antibiotics for a variety of bacterial infections. Microsurgery. Remedies for childhood poisoning. Management of epilepsy. Cystic fibrosis. Organ transplantation. Correction of congenital heart defects, etc. All this is possible only because of the animals. So without animals, newborns who develop jaundice each year would contract cerebral palsy. Now it is preventable. There would be no kidney dialysis. A surgery of any type would be rare and extremely painful because there would be no anesthesia, which has been eradicated, would continue unchecked. Dogs, cats, other pets, and farm animals would have died from anthrax, feline leukemia, rabies, and more than 200 deadly diseases, which are now preventable. So these are the small animals which are being used in the research. I should not mention the names. But why are animals used in research? We all know that the organs and body systems are similar to humans and other animals. and they are susceptible to the same diseases that affect humans. Very close relationship. Short lifespan that allows the animals to be studied throughout their entire life. To keep experimental variables to a minimum. A Russian scientist kept the dog alive for few, which was beheaded, and this was the breakthrough at that time. The animals experimentation is required before it can be passed on separately to humans. Of the laboratory involves two most important responsibilities on the part of the experimenter. First, the animal is handled with utmost care so that it does not suffer any pain. And secondly, a due regard to the health and well-being of the animals colony. Even when they are killed in of experiment, it should be done by a human method. We call it as the euthanasia, painless killing. And there are a number of techniques for this. And nowadays, normally, gaseous or anesthesia used for this purpose. And this is, we must take care of these animals. And this is the ethics. One should follow the guidelines. One should take care of these animals. If you see the history of ethical committee, the first animal rights laws were introduced between 1635 to 1780s. And in 1635, Ireland was the first country to pass animal protection legislation, which was related to an act against ploughing by the tail and pulling the wool off from the living sheep, because this was very painful condition. And Ireland was the first country. In India, the first war before independence started with the issues of animal welfare in 1857. Since then, various laws were passed as an act, like in 1857, 1859, 1861, 1867, so on. It is 679, 1889. This, about 150 years, the laws were passed. They were not properly enacted. The Prevention of Cruelty to Animals Act 1890, see, about 130 years back, it contained a clause providing a penalty for killing any animal in an unnecessarily cruel manner. But a blanket was given. That was exception, the rights on use of any race, tribes or class. So in 1890s, there was a law. The Madras SPCA made an effort to arouse interest in the use of human killers. Srimati Rukmini Devi Arundale, a nominated member of Rajya Sabha, introduced the Prevention of Cruelty to Animals Bill in 1953. And Pandit Jawaharlal Nehru assured the House to form a committee to look into the matter. Subsequently, the bill on PCA Act 1960 was passed and enacted. The Animal Welfare Board of India, AWBI, was established under the Ministry of Food and Agriculture and started functioning during 1962. See the situation in 1962, it was under the Ministry of Food and Agriculture. Now it is under the Ministry of Fisheries. They have the AWBI CPCCA is now governed by the Ministry of Fisheries. Before that, it was under the Ministry of Environment, Forest and Animal Welfare. The awareness of abuse and exploitation of animals by humans is called as the animal welfare. By maintaining appropriate standards of accommodation, feeding, and general care. The prevention and treatment of diseases and the assurance of freedom from harassment and unnecessary discomfort and pain. This is animal welfare. The AWB is presently functioning under the Fisheries Government of India. PCA it was enacted in 1960. Section 15A of PCA, that is contains the Committee for the Purpose of Control and Supervision of Experiments on Animals, CPCCA. It is the statutory body of Government of India, and it was first constituted in 1964. In 1996 with 27 members, and chairperson was Mrs. Maneka Gandhi. And from here, new era related to animal care started. And much has been done by the Government of India to have related to this animal welfare. Now CPCC is mandatory for every institution which is working in the area of animals or carrying out research. This is made mandatory. Breeding of and experiments on animals were introduced in 1998, and the IAC implement these objectives. Given by the CPCCA since 2001, and the latest is being given in 2000, or it is given in 2018. These are the latest guidelines. Majority remains same, but certain changes has been done according to the requirement. Now this is the website of the Committee for the Purpose of Control and Supervision of Experiments on Animals. This is CPCCA. It is under the Ministry of Fisheries, Animal Husbandry and Dairying Department of Animal Husbandry and Dairying Government. Just before few slides, I told that initially the CPCC was under the Ministry of Agriculture. Then it was shifted to change, and now it is under the Ministry of Fisheries. This was one of the reasons of poor implementation of guidelines by of the CPCCA. But since last 10 or 20 years, CPCC is functioning well, and the guidelines are being implemented properly. So CPCC is National Level Expert Committee that scrutinize and approve the use of large animals in research. Introduce a National Good Laboratory Practice document that was not available before. National Committee for the Promotion and Preparation of the Concept of Alternative in Education, Basic Biomedical Research and Regulatory Testing was introduced in 2000. And it has made mandatory registration of animal laboratories with the CPCC. UGC, AICT, Pharmacy Council of India, they have all issued guidelines that the educational institutions who are using the laboratory animals, they should mandatorily have registration with the CPCC. The number of registered institutions was very less around 2000, and it has now increased. Constitution of Institution Animal Ethics Committee, that is must under this CPCCA, that is the institutions who are using animals, they should have their own IAC. CPCCA nominates the nominee for all these committees. Good practices, that is GLP, this is required for quality maintenance of and safety during conduct of experiments. The goal of these guidelines is to promote human care of animals. Again, I want to give the emphasis is related to the promotion of human care of animals. The animals should be treated humanly. Composition. There are a number of members in IAC. The 5 members which are nominated by CPCCA by the institution, 1 biological scientist, 2 scientists from different biological discipline, 1 veterinarian, 1 scientist in charge of animal facility. Other members are nominated by CPCCA, main nominee, link nominee, scientist from outside the institution, 1, our member. So objectives of the IAC is to authorize and approve the experiments on small animals, only small animals. Remember, IAC is authorized to approve the experiments on small animals only. For larger animals or for use of primates, it should be forwarded to CPCCA. IAC is not authorized to approve the experiments on animals. All experiments should be carried out under the supervision of qualified individuals. Pain-causing experiments should be avoided or should be done under the anesthesia. And wherever possible, the experiments on animals must be avoided. But as far as possible, the small number of animals should be used in the experiments. Experiments on large animals be avoided when it is possible to achieve similar results in small animals. Experiments are not performed solely to attain manual skills. Even the repetition of the experiment is not allowed by IAC or CPCCA. if a dose of the drug, for example, any extract or plant material, which is already being used by the scientist or it is published in the research paper, it is not necessary that the person should again decide the dose or perform the toxicity studies on animals. We can use that data and we can use that dose in our experiments. But proper review of literature should be done for this purpose. Animals under experiments are properly looked after as per the CPC's guidelines. There should be proper care of the animals. Suitable records on animals issuance, use, and sacrifice for each experiment must be maintained. The function of IEC is to review and approve all the types of research proposals involving small animals, and this should be done before the start of the experiment. Visit the animal facility and laboratories and provide reports. Earlier it was 3 years, now it is 5 years, and is reconstituted at the time of renewal of registration. One should maintain complete confidentiality, and that should be maintained. There should be proper maintenance of the record. A minimum of 6 members are required to form a quorum. Presence of nominees must. Without nominee, the committee or the IAC is not valid. If nominee is not able to attend the meeting or is not able to the link nominee is can attend the meeting, but that should be approved by the main nominee. Socially aware member should be there for at least one meeting in a year, and this should be referred to the CPCC. Decisions in meetings are done by majority and not by circulation. Proper records should be maintained by the IAC. Form A related to the registration of establishment. Form B related to the animal experiments protocol. Now it is slight modification. This Form B has been slightly modified. New Form B is available on the website of the CPCCA. Earlier, a number of animals which were being used by the experimenter was not mentioned in the certificate. Now this has been modified, and one should mention the number of animals which are being used or which are being sanctioned for the particular protocol. Form C, all animals procurement and insurance records should be maintained by the animal facility. And Form D, this for the investigator. Investigator must maintain the record of use, the number of animals sacrificed, record of rehabilitation of animals should be maintained. Rehabilitation particularly is done for the large animals, and their proper protocol should be followed. In case of dead animals, postmortem reports are required. MO of all IEC should be maintained. Animals breeding, issuance, sacrifice, and mortality records should be maintained by institution. Environmental conditions like temperature, light, and sound records should be properly maintained. Animal training and attendance should also be maintained by the institution. and the institute should ensure during the holidays and record euthanasia and disposal. Environment parameters should be maintained and monitored. There should be 100% share. Liquid waste should be treated properly. Solid waste should be properly treated. There should be complete and proper record. There should be maintenance of SOPs, etc. All these are guidelines which are given by the CPCC regarding the temperature and humidity control. There should be proper sanitation. There should be proper food given to the animals. There should be proper bedding that should be neat and clean. Potable water should be provided. I discussed about the 3R principle, that is reduction, refinement, replacement. Now there is a fourth principle, and this should be mandatorily implemented in the animal house. Wherever possible, the number of animals, the use of animals should be reduced. If the data is obtained in 3 animals, then 3 animals should be used. Repetition should be provided. Animals which are required for the experiments, that should be procured from the registered breeder only. CPCCA gives approval or registration related to the experiments or breeding or trading. Those animals are registered with CPCCA. From these animals, the animals should be procured. And this, after procured, they should be, and then they are allowed to stabilize, and then only they are used for the purpose. The duration of experiment, the maximum permissible time for the experiment is 3 years. But in some cases, it can be more than 3 years. See, the life of the animal, small animal, is very less. And that is the advantage for experimenter, because the whole life cycle can be used for the experimentation purpose. And that's why the IAC or CPCC has given this. But if the life is more, then and it is required, if the data is required, then the permission can be given for study more than for 3 years. These are the guidelines which are given by the CPCCA, and these are the major guidelines that should be followed by everyone. There are other guidelines also which we follow. One is the ICH, that is called International Conference on Harmonization, and this was created in April 1990 at Brussels. It involves the regulators and industry for discussing scientific and technical matters to ensure and assess quality, safety, and efficacy of drugs. We also follow these guidelines. The guidelines were passed with the aim to achieve greater harmony in the interpretation and application of technical guidelines. It is also subjected to reduce duplicacy of testing during research and development of new drugs. This is important. Duplicacy. Unnecessary research activities which are duplicate, that should be avoided. An international conference on harmonization provides these guidelines. One should avoid this. Structure of ICH. There are members from regulatory body like European Union, Ministry of Health, Labor and Welfare, Japan, US, FDA, and from industry like EFPIA, JPMA, or Pharmaceutical Research Manufacturers of America. These members, they constitute the ICH. Another guidelines which are related to the studies on this is the OECD guidelines, Organization for Economic Cooperation and Development. These OECD guidelines, particularly these guidelines are used for studying the toxicity of the drugs. It includes testing of chemicals. And this testing should be in accordance with the physicochemical effects on biotic system, environmental properties, health effect, and other areas such as pesticides. And these guidelines are internationally accepted. These are the standard methods. Particularly, most of the researchers, they use these guidelines for toxicity studies. Particularly the fourth number series, OECD guideline 423, OECD guideline 419, 427. Initial, particularly the fourth number, the guide 423, 425, 427, that is particularly related to the biological system. And these guidelines are used for carrying out toxicity studies or deciding the dose. Wherever possible, one should minimize the use of animals or should prevent the use of animals. We can use the alternative methods for this purpose, like flies can be used, tissue culture can be used, we can use vitro models, we can use the in silico modeling or softwares. So my aim here was to provide discussion on the use of animals, and these animals, they should be used in a proper, they should be, one should follow the proper guidelines given by the CPCC, ICH, and wherever possible, one should use alternative methods for carrying out the research. Thank you. Over to Prashant. Am I audible, Prashant?
Yes, yes, yes. Should I move forward, sir? Should I move forward?
Yeah.
Okay. So thank you so much, sir, for sharing a valuable insight in the topic use of experimental animals in research with special reference to CPC, SEA, and other guidelines. So the topic remains of great interest, and afterwards significant debates, number of debates, UGC has proposed a partial ban on this topic, use of experimental animals in research. So in 2011, UGC has proposed a partial ban, apex body of University Grant Commission. They have proposed a partial ban in 2011, And afterwards, further review, they have imposed in 2014 a ban on both UG and PG studies.
Yeah.
So now I'm going to invite the chairperson of the session, Professor B.K. Bastia, sir, Head Department of Forensic Medicine, AIMS Rishikesh, to put some light and concluding remarks of the session. Over to you, sir.
Is it okay? Am I audible?
Yeah.
Okay, sir. Thank you, Dr. Surendra sir.
Thank you, sir.
आप इतने extensively cover किए, so I don't have anything to add actually, frankly speaking. But I will tell something. Let us revisit the ethics, morality, and science on this perspective, sir. When we talk about cruelty and misuse of animals, जब हम बात करते हैं, वहाँ पे हम एक नेगेटिव कॉनोटेशन ले आते हैं. क्रूएल्टी, मिसयूज, पेन, टॉर्चर, वो सब ले आते हैं. Actually, I have a book here.
Yeah.
The name of the book is, can you read it?
Just a minute, sir.
Ethics by Benedict Spinoza.
Yeah.
Can you see it here, sir? Here.
Yeah. Ethics. Yeah.
मैं पढ़ चुका हूँ. वहाँ से मैं थोड़ा बात करता हूँ, थोड़ा.
नहीं जैसा बिल्कुल.
He has analyzed the ethics in a different perspective, sir.
Yes, sir. Yes.
वो क्या बोलते हैं? वहाँ पे लिखे, do the animals, do they have mind? First वो एक analyze. आत्मा उनका होता है, वो भी analyze किए, और भी लिखे, can they think? Do they think? अगर हम सोचते हैं, हम वो लिखे हैं यहाँ पे, when we think, we always think using some words.
Yeah.
अगर हम सोचेंगे कुछ, बिना वर्ड यूज किए सोच नहीं पाएंगे.
Yeah.
ये किताब में एक्सटेंसिवली दिए. दिस इज ए बिग, मतलब वॉल्यूम, एक वॉल्यूमिनस. सिर्फ इसका ऊपर है. सर, आई थिंक यू मस्ट हैव रेड इट. मैं उसको और कुछ बताऊंगा आपको, और एक किताब के बारे में, जब साइंस, वहां पे ये सब दिए. एंड वहां पे लिखे, व्हाट ही हैज रिटन देयर, वी थिंक ऑफ ओनली, वी टॉक ऑफ दोज एनिमल्स. हम उस एनिमल के, जानवर के बारे में बात करते हैं जिनका पोजीशन में हम खुद को रख पाते हैं. हम एक डॉग हो या लाइव एनिमल के बारे में जब सोचते हैं, हमको वो उनका पोजीशन में रख के वहीं पे हम एथिक्स का बात करते हैं. जबकि हम छोड़ जाते हैं क्या, जैसे कि प्लांट का भी, अगर उनका भी सोल है, then the plants and bacteria, they also have soul.
Yeah.
Beautiful analysis they have done, sir. And then if that is so, then what about the antibiotics? What about our food?
Yeah.
So I request all of you, all everybody, to please go through this book. It is a very, not that much costly book. Ethics by Benedict Spinoza, Spinoza. Ethics by Ben. अगर हम ये किताब देखेंगे तो our the concept on the ethics, it will change radically, sir.
Yeah.
Okay.
Yeah,
so I request all of you to go through this one. The second is, the second point that I want to add is the science.
Yeah.
This is morality. Morality के बारे में बात कर लिए, cruelty, ethics का science. The first question is the concept of GMO, genetically modified organisms.
Yeah.
And whether whatever we are using, are they genetically compatible? Whether do those animals, they respond to a disease or an antigen or pathology or whatever whatsoever in the same way as we do.
Yeah.
There is another beautiful book written by Robert Sapolsky. Please check this. Please write the spelling. This is a very
title.
This is a free book available on net.
What is the title of book, sir?
Sapolsky. written in 1994. वहाँ पे ये लिखे हैं वो, even though the antigen, antibody, receptor, immunity, everything is same. Everything same. हमारा ह्यूमन का उनका सेम है.
Genetically modified.
Dynamics is different, sir.
Yes, sir.
Dynamics के बारे में लिखे हैं कि वो किताब में लिखे, बुक का नाम है Why zebras, the striped animals, why zebras don't get ulcer. उनका अल्सर क्यों नहीं होता है?
Don't get ulcer. Right.
तो एक किताब का इंट्रोडक्शन ऐसे किया गया है. हमारा तो डेंजर है ही नहीं कुछ.
हाँ.
उनका तो डेंजर है ही ना ऑल द टाइम.
Yes, sir.
कोई एक लायन आके अटैक करेगा, वो करेगा.
They are always under stress.
क्यों होता है? वहाँ पे वो लिखे, हम जो human beings, we live in the sympathetic system. Sympathetic nervous system हम वहाँ पे ज्यादा जीते, हमारा माइंड को ज्यादा यूज करते. वो लोग उनका जो parasympathetic nervous system है, parasympathetic nervous system, that is the healing nervous system. हमारा दो पार्ट होता है ना, एक sympathetic,
parasympathetic. Right, sir.
वो जानवर जो जेब्रास, एनिमल्स, जो भी आप बात कर रहे हैं, वो parasympathetic mood में जीते हैं वो.
Yes, sir.
उनका response is totally altogether different from ours.
Yeah.
उनका जो
parasympathetic system is dominant.
एक response to a pathogen that is altogether different in the animals. वो तो खाते, हम तो बैक्टीरिया को देख के खाते हैं, वो तो ऐसे ही खाते हैं वो. They never eat the cooked food.
This is actually animal variation, sir.
Yes, sir.
Yes.
हम जो एक्सपेरिमेंट कर रहे हैं, is it really simulated? वहाँ पे ऑपरेशन किए हैं वो रॉबर्ट सैपोल्स्की.
Similar to, sir, actually rats. If you see the rats, they lack vomiting center, and therefore we cannot relate to vomit.
हम जो एक्सपेरिमेंट करने जा रहे हैं, हमको ये रिविजिट करना है.
Yes, sir.
हम जो कर रहे हैं, यस क्रुएल्टी है तो ठीक है. एक्चुअली क्या होना चाहिए? फर्स्ट जो पॉइंट में बोला, लव, कंपैशन एंड एम्पैथी. ये लैंग्वेज में सोचना चाहिए rather than cruelty, misuse, sacrifice. ये सब नहीं है. हम जैसे हमारे बच्चे को बिहेव करते हैं, we should treat the animals, lab animals like our kids. वो एक है. सेकेंड पॉइंट जो दिया है यहाँ पे रॉबर्ट सैपोल्स्की की किताब जेब्रास डोंट गेट अल्सर, वहां पे लिखे हैं कि हम जो एक्सपेरिमेंट करने जा रहे हैं, whether it is really simulated, वो हमको सोचना है. बिकॉज our response and their response, that is different. So हम ये दोनों सोच पाएंगे तो एथिक्स ठीक है, बट साइंस के बारे में ये अगर सोच पाएंगे तो we can really advance forward. Otherwise we can हम क्या कर सकते हैं? हमारा जो GMO, आजकल तो बायोटेक्नोलॉजी आ गया, हमारा वहां पे भी हमारा जीन देखे, जो भी है लाइगेज पैगेज, जो भी है रिकॉम्बिनेंट DNA टेक्नोलॉजी से हम देखे, हम रियली बढ़ सकते हैं आगे. सर thank you all the organizing.
Thank you, sir. Thank you very much.
Thank you so much.
Thank you very much.
Thanks for your suggestion, sir.
Yes, sir.
गैतोरी सर माइक ऑन कर लीजिए. माइक, योर माइक.
I know. So thank you once again, Bastia sir, for sharing these valuable things. And now we are inviting questions from our gathering. So if you have any questions, you can ask. You can ask questions in dual mode. You can ask directly, or you can share your chat with us so that you can ask questions on your behalf. Any questions from the gathering?
गैतोरी सर, मेरा एक क्वेश्चन है. सर, what के सर?
Yes, sir. Go, sir.
So, just like a lot of guideline was coming from the different agencies that you don't do this, don't do this, don't do this on the animals. But if you compare the research in India and Pakistan and other country, and if you compare that research nowadays, lot of research is there. In Pakistan, they are doing the research in the graduation also on the animals, but we are our pharmacy council abolished all the
So this is actually one should think on this, because if you are not handling the animals at undergraduate level and you are directly coming to PG, which the problem which we are facing, the students who are coming from pharmacology, they are not aware of any or the use of animals, or even about, they don't know the difference between the rat and the mouse.
Yes, yes.
This is the problem. If you are not using at the undergraduate level, but actually these are the guidelines which are given by the government of India, which are mandatory, and we have to follow, because UGC, AICT, all these regulatory bodies, they have issued the guidelines. But here one should think on that we should allow the use of animals, particularly for pharmacology purpose, and that should be regulated properly.
Problem is that a lot of agencies, council, they are just avoiding on the behalf of human rights or on the behalf of animal ethics. But problem is that after
they are avoiding, this is the proper word, actually they are avoiding, they want to save their money.
Yes, sir.
But there are a number of things without animals you can give the knowledge to the students, because for example, we are using the isolated pork ileum preparation.
But simulated system is not of that perfect. You cannot compare
it's actually, sir, it is not simulation. It's, if you take the ileum, it is exactly similar to the rat. We can obtain it from the slaughterhouse, from the pork ileum. We can perform number of experiments, but these are the limited things.
अभी भी मेरे को याद है जब मैं ग्रेजुएशन में प्रैक्टिकल्स करते थे तो हमारे टीचर्स भी they are forcing that you have to sacrifice the animal.
Right. Yeah.
But nowadays nobody is interested in doing the practical in the laboratory. They have just make the museum of the pharmacology laboratory.
This is actually
after 20 years. My question is that after 20 years, what will we will do in the pharmacology? That will be a third.
Yeah.
Forensic.
Actually, this is the disadvantage.
Pharmacy.
This is actually the disadvantage.
Yes, sir.
If persons are not even trying to use the alternatives which are available. I am saying that if you use the pork ileum, it will provide the exact result. Means it is not scientifically valid result, because it is not repeatable. But you can use this as a demonstration tool to the students. But teachers or even the institutions are not trying to use these methods.
Yes, ठीक है sir. Prashant sir, please.
Okay, thank you so much, sir. I'm requesting all the participants to turn your camera on for a group photograph. All participants. Dr. Ashwini Kumar, all the participants for a formal group photograph of the session. Yeah. Is it fine, Goel sir?
ठीक है sir. Thank you. Thank you, sir. Thank you for connecting with us. So, on behalf of National Science Day Organizing Committee, I welcome to all person. Vinay sir, we are very much thankful that you give some time to the Gurukul Kangri University. Thank you, Prashant sir. Thank you.
Thank you.
Thank you very much, Prashant sir. Now, in continuation, I request a presentation by Silky Sethi. If she is hearing me, she can join or she can start her presentation.
Good afternoon, sir.
Good afternoon. Welcome to the platform of Gurukul Kangri Vishyavidhyalaya, Aushdipad of MahaKumbh, celebrating National Science Day.
Thank you, sir. Thank you, sir.
Please continue with your introduction.
Yeah, sir, I'm Silki Sethi and I'm continuing my PhD from MIT University, Noida. And I will be delivering a presentation on tetrazoles today. Can I share my screen, sir?
Please
Sir,
share.
okay, sir. सर आप मुझे परमिट कर दीजिए प्लीज अलाउ मी टू शेयर द स्क्रीन
You are already permitted actually.
Sir, I'm not able to share my screen actually.
Okay, by which ID? The same ID?
Yes, sir. Silky Sethi. I've logged in through Silky Sethi 1981@hotmail.com.
Actually, from my side, it is already you are presenter actually. I made you presenter. Yes, yes. Your screen is now visible.
So my screen is visible.
Yes, yes, it is visible.
Okay, sir. Good afternoon, everyone. I'm working in K.R. Mangalam University and I'm doing my PhD from MIT University, Noida. So one of my topics is significance of tetrazole moiety in angiotensin 2 receptor blockers. I've been working on angiotensin 2 receptor blockers. So I have opted one of the moiety, which is tetrazole moiety, which is present in angiotensin 2 receptor blockers. So tetrazoles, I would be talking much more about tetrazoles today. Tetrazoles are basically, this is a 5-membered ring containing 4 nitrogen atoms and 1 carbon atom. And it is, tetrazoles belong to the class of 2 unsaturated 5-membered rings having aromatic tetracycle consisting of 1 carbon and 4 nitrogen atoms. So basically has 2 double bonds in it with the 4 nitrogens and 1 carbon in it. And the most direct and versatile method for the synthesis of tetrazoles is the cycloaddition reaction, which is that is 2+3 cycloaddition reaction between nitriles and azides. Tetrazoles, if we come to the substitution, of tetrazoles. Tetrazoles can be classified as non-substituted tetrazoles, that is unsubstituted tetrazoles, mono-substituted, di-substituted, and tri-substituted tetrazoles on the basis of the number of substituents which the tetrazoles are carrying. Then if I talk about in the substitutions, 5x substituted tetrazoles, this is quite prevalent and it exists in 2 tautomeric forms, that is form 1 and form 2. Basically, tetrazoles were discovered by Bledin over 120 years ago, and this has gained a lot of importance because of the kind of this pharmacophore which it has. It is a very flexible group and it can bind to various receptors. So it is quite, the number of publications on tetrazoles have increased because it is an important group for it has versatile therapeutic potential, and particularly as in antihypertensives, it is an important group for the antagonistic activities of antihypertensive, primarily of angiotensin receptor blockers. So tetrazole has, apart from antihypertensive activity, tetrazoles have antibacterial, antifungal actions. So this is about the versatility of the tetrazoles, that the tetrazoles are very stable heterocycles. When it comes to stability, tetrazoles have been very stable. When we come to pentazoles, which is one more nitrogen, pentazoles are very unstable. When we compare tetrazoles to pentazoles, pentazoles are very unstable and they have been found to be highly explosive. Even at low temperature, they have been explosive. Whereas tetrazoles are very much thermally, they are very much stable compounds. And as we have already discussed, I have already told that tetrazoles have broad applications. So it has been into medicine, it has applications in medicines, it has applications in biochemistry, in explosive industry, in pharma. Yes, sir. Sir, am I audible? Am I audible?
Yeah, ma'am, you are audible.
Okay, sir. So when it comes to various industries, tetrazole derivatives have been used, and many of the medicinally important compounds which have been approved by US FDA contain tetrazole moiety. When we come to anti-cancer drugs, they have tetrazole moiety. If I talk about letrozole, it is an anti-cancer drug which has tetrazole moiety. When we come to antibacterials, we have ceftizole and cefamendole, which have, these antibacterial properties, and this has tetrazole moiety in it. So why we are taking tetrazoles? Because tetrazoles it is a kind of a group which provides basically prolonged half-life to the drug, and when it is added to the drug it has more metabolic stability as well. If we talk about we compare COH group to tetrazole group, so the drugs having COH group, although both of them are bioisosteres, but then tetrazole does not go through, these drugs drugs containing tetrazole do not go through go through the metabolism, liver metabolism. So basically they are more metabolically stabilized, and also tetrazoles have a better membrane potential. So that is why, since they have more membrane potential, they have been used to improve the oral bioavailability, and they have increased lipophilicity as well. And that is the reason tetrazoles have been used these days as drugs. I would like to come to the angiotensin receptor blockers. As we know that in antihypertensives, ACE inhibitors and angiotensin receptor blockers are the two primarily main drugs which have been used as antihypertensive agents. ACE inhibitors have not been used much because they have been used, but then they have certain kind of adverse effects, such as they have been causing cough, they have been causing oedema. So angiotensin receptor blockers are more used these days, especially to those patients who cannot tolerate the ACE inhibitors. So we have been using ARBs instead of ACE inhibitors. And when we come to ARBs, there are two types of ARBs, that is the peptide ARBs and non-peptide ARBs. Peptide ARBs are very less frequently used as compared to the non-peptide ARBs, because the peptide ARBs have low bioavailability. The major adverse effect of peptide ARBs is that it has low bioavailability. Also, the duration of action of the peptide ARBs is short as compared to the non-peptide ARBs. That's why we have shifted to the non-peptide ARBs, and now I'm talking about the non-peptide ARBs, the pharmacophore of the non-peptide ARBs. So the ARBs, generally when we talk about the non-peptide ARBs, basically the three major rings which are present in the non-peptide ARBs is imidazole ring, then the biophenyl ring, and the tetrazole ring. These three rings, groups are important, although tetrazole ring has been replaced with certain other groups such as carboxylic group also. But yes, in major ARBs, these three rings have been important. They have played an important part. If I say imidazole ring, so imidazole ring has been binding to the amino acids in the helix 7. helix 7 of the angiotensin receptor, because all these ARBs, they have been blocking the mechanism of action of these ARBs, as we all know, that it blocks the binding of the angiotensin 2. The main objective of ARBs is that it has been blocking the binding of angiotensin 2. So basically the ARB which are been blocking the binding of angiotensin 2, the imidazole ring present in the ARB binds to the amino acids present in the helix 7 of the angiotensin receptor. The biphenyl ring binds to the amino acids of helices 6 and 7, and the tetrazole group, which is present again in the ARBs, it interacts with the amino acids present in the helix 4 and 5 of the angiotensin receptor. Thus, these three rings play an important role in preventing binding of angiotensin 2, and they themselves bind to the receptor. And also, it has a tetrazole. When we incorporate tetrazole group to the angiotensin receptor blockers, the binding of the drug is enhanced. It has a very high binding affinity for this angiotensin receptor, AT1 receptor basically. Also, tetrazole has been a bioisosteer to carboxylic acid. Basically, bioisosteers are the functional groups which have similar physical properties and chemical properties. And these similar physical and chemical properties, such as similar physical and chemical properties, se meaning hai ki same electronic environment and similar size and similar comparable pKa. So tetrazoles have been bioisosteers to carboxylic acid group, and as the slide shows that it acts as a bioisosteer to the carboxylic group, and all tetrazoles act as bioisosteers to carboxylic groups. And the reason behind this bioisosterism is that the mobile hydrogen of the tetrazole has a comparable pKa and have similar size basically, and special arrangement of the electrons, and the electrostatic potential is also same. So that is why the carboxylic acid and tetrazole groups both are bioisosteers to each other. That is why tetrazole can be replaced with carboxylic acid in the angiotensin receptor blockers. So if we come to the binding properties of tetrazoles, tetrazoles have a very good binding properties, and if we talk about why the tetrazoles are binding good nicely to the angiotensin receptor, the reason behind is that there are a lot of nitrogens in it. If we go to the slide 1, we can see that here 4 nitrogens are there, and because of the high density of nitrogens present in the tetrazole, that high density of nitrogen which is present provides a lot of opportunities to form hydrogen bonds and with the receptor sites, and because of this kind of hydrogen bonds which are formed with the receptor site, the binding affinity of tetrazoles is increased. And basically tetrazole is an acidic group, so when it binds to the basic portions of the receptor, basic positions, the basic groups of the receptor. Since it itself is acidic, so tetrazole is generally present at the ortho position of the biphenyl ring. This is I can show the structures here. So this is the ortho position of the biphenyl ring. It is generally present at the ortho position of the biphenyl ring, and when it binds to the receptors, since it's an acidic group, it binds to the basic portion of the receptor, the basic groups present in the receptor. So tetrazole at the first position of the biphenyl methyl group binds to the basic positions in the receptor, and thus it serves as an important group. It serves as a very important group for the potent antagonistic activity of the angiotensin receptor blockers. So ARBs, as I've already discussed, that ARBs have been exerting their action by blocking the binding of angiotensin 2 to AT1 receptor. Two types of ARBs, peptide and non-peptide, I have already told. And then we come to the derivatives, antihypertensive drugs which have biphenyl tetrazole derivative, which have the tetrazole rings in them, that are losartan, irbesartan, candesartan, valsartan. Losartan was the very first approved drug, which was a non-peptide drug, and it was orally active, and it had antihypertensive properties in it, and it specifically binded to the AT1 receptor subtype, and it was the very first approved non-peptide drug having the tetrazole moiety in it. So the various tetrazole containing drugs, losartan, valsartan, they have been used in the treatment of hypertension, and they are into the clinical practice. They have been used, they have already been used clinically, and thus these tetrazole moieties have been showing a lot of potential. These tetrazole derivatives have been showing a lot of potential as a putative antihypertensive drugs. So this is the mechanism of action, how these ARBs have been exhibiting antihypertensive activity. Losartan prevents the blood vessels from narrowing and improving the blood flow. And as it has been the very first antihypertensive agent. Also, apart from antihypertensive agent, it has been used in heart failure also. And losartan has replaced ACE inhibitors. It has been used by all those patients who were developing cough with ACE inhibitors. So losartan was the first orally non-peptide ARB. And then the valsartan, then came gradually the valsartan, irbesartan, candesartan. These are again ARBs, angiotensin receptor blockers, which have been relaxing the blood vessels, and they have been blocking the angiotensin 2, and we all know that angiotensin 2 has a vasoconstrictive action, so they have been preventing the angiotensin 2 from binding. Now, if we come to irbesartan is again a very, very prominent drug as an anti-angiotensin receptor blocker, and it has a high bioavailability. And if we talk about, we compare the pharmacokinetic properties of the various sartans, we can say that losartan, these are the 5 drugs which have tetrazole moiety in it. That is, losartan has tetrazole, valsartan has tetrazole, irbesartan has tetrazole, then candesartan and olmesartan also has tetrazole. Highest bioavailability is of irbesartan, that is 70%, and iprosartan has a relatively low bioavailability. But then high bioavailability of irbesartan is because of the reason that it has a cyclopentyl group present in it, which binds to the receptor very firmly, and because of which this irbesartan has a high bioavailability. The cyclopentyl group which is present in irbesartan forms hydrophobic interactions with the hydrophobic pocket of AT1 receptor. So this kind of hydrophobic interaction with the hydrophobic pocket of AT1 receptor makes irbesartan a very high efficacious drug. That is, its bioavailability is enhanced because of this particular cyclopentyl group. And in this particular slide, we can see the various sartans which have basically tetrazole groups. Here is the losartan. Losartan again has a tetrazole group. Irbesartan has a tetrazole group. And I was talking about this cyclopentyl group. This is again a strong reason for the high bioavailability. Apart from tetrazole, also plays a major role in increasing the bioavailability. But then this cyclopentyl group also forms hydrophobic interactions. Then comes the valsartan. Valsartan also has a tetrazole group. But then telmisartan is the drug which does not have a tetrazole group, and since tetrazole and carboxylic acid are bioisosteres to each other, that is, they exhibit similar biological properties, and since they have similar physical and chemical properties.
Excuse me.
Yes.
Can you make it fast, please?
Yeah, yes, sir.
Slide fast.
Okay, sir. So telmisartan is again, it has a COH group in it, so that is why it has lesser. It is being replaced with the tetrazole group. So this is about candesartan. Candesartan also has the tetrazole group in it. This is the various pharmacokinetic parameters which are there in the various drugs. This was almost the last slide, sir. These are the references, and thank you, sir.
Thank you, madam, for joining us.
Sir, do I need to, there's some questions or something?
Yes. If anybody, the session is open for question and answer. So if any participant is having any query, they can ask directly or they can type in chat box. So I think the presentation was so nicely presented that all the participants have got full information from this presentation. Thank you very much, Silky ma'am, for joining us.
Thank you, sir. Thank you.
So in continuation, I am very thankful to our audience that they are continuously connected on this platform of Gurukul Kangdi Vishwavidyalaya. Now in continuation, we will continue with the industry academia interaction session. And in this session, the eminent speakers are joining us, like Professor Kanchan Kohli from Jamia Hamdard University, and Dr. Hem Joshi, who is from HR Solution Consultancy, Haridwar, and Dr. Neeraj Sharma, who is a team leader at CoScience Med Tech Company Limited, Beijing, China. Another eminent speaker in this interaction between academia and industry is Sunil Bhatt, who is a general manager at Sanjeevani Parenteral based Dehradun. Dr. Amit Bhatia will be joining us, who is from Department of Pharmaceutical Sciences and Technology, Maharaja Ranjit Singh Punjab Technical University, Bathinda, and Dr. Ashutosh Shukla, who is a senior clinical research scientist and a team leader at Drug Discovery and Development, Patanjali Research Institute, Haridwar. So with the blessings of our chairman, sir, of this event, Professor R.C. Dubey, sir, our convener, Professor S.K. Rajput, sir, our organizing secretary, Dr. Vipin Kumar, sir, and Dr. Ashwini Kumar, coordinator of this session. I'm also very thankful to our sponsoring agencies. This session or this event, I must say, is sponsored by Uttarakhand State Council for Science and Technology, UCOS Dehradun, and we are also thankful to State Medicinal Plant Board, Uttarakhand, who has supported us for conducting this National Science Day during Ashdiya Padak MahaKumbh 2021, celebrated by or organized by Department of Pharmaceutical Sciences, Faculty of Medical Science and Health, Gurukul Kangdi Vishwavidyalaya. So once again, I welcome you all in this industry academia meet. So today's session will be started with the knowledge-based sharing from Dr. Hem Joshi, who is HR Solution Consultancy, Haridwar. I request our colleague, Dr. Vipin Kumar Sharma, who is also organizing secretary of this event, to please introduce him in a short words and invite him for his deliberation. Over to Dr. Vipin Kumar, sir.
Thank you, sir. Thanks a lot. Am I audible?
Yes, sir. Yes, sir.
Please, audible.
Thank you, sir. Namaskar and good afternoon to all the participants, all the dignitaries, all the guests who are connected on this online mode. And some of the participants are also available in the department that are joining this event by offline mode. So first of all, I must say thank you to our dignitaries, to our administration, to our Honorable Vice Chancellor, Professor Rup Kishore Shastri ji, who supported, who motivated us to take this opportunity for organizing Padak MahaKumbh with the theme of MahaKumbh being organized in Haridwar in style. And our registrar sir, Professor Dinesh Bhatt, an active, eminent scholar, academician, and under his supervision, our Visvidyalaya is getting more accreditation. And our Dean sir, Professor R.C. Dubey, who is a well-known figure in the field of microbiology. And not in microbiology, he is also a well-known figure in spiritual science also. So thank you, sir, for giving us this opportunity to organize this event, 2 days event. Our head sir, Professor S.K. Gupta ji, S.K. Rajput ji, Kumar Rajput ji, and is a person having the leading capacity, and under his leadership, day by day, we are getting new achievements. Thank you, sir, for giving us this opportunity to organize this 2 days event. So today we have numbers of participants and eminent personalities from industries. As our coordinator of the session, Dr. Prince Prashant Sharma has told that we have M. Joshi sir with this event. M. Joshi sir is post-graduated in Human Resource Management from MIT Ghaziabad, and he is a HR professional with working experience of more than 27 years. He has worked with pronounced companies as HR head in the sectors of office automation, telecommunication, and pharmaceutical industry like Pfizer, Modi Ji Rocks, SR Cellphone, Reliance Communications, RFCL, Joitis, Zydus Cadila, leading HR operations in corporate, employee engagement, team building, merger, acquisition, HR policies and processes, HR audits, training and development, and so on. So number of responsibilities have been completed by M. Joshi in different capacities. So such personality we have in this session, in Industry Academy session. So sir, once again, I would like to invite you in this session, sir, for this Industry Academy meet. And we are very much fortunate to have you, and all of us will certainly get benefited with our motivational talk. So with these words, I would like to invite you, sir, in this session. And in this session, we have with us Dr. Asutosh Shukla ji from Patanjali Research Institute, Patanjali. Dr. Shukla is associated with clinical trials in Patanjali. As we know, a well-known figure in developing the herbal products by Baba Ramdev ji and Acharya Balkishan ji. So with the blessings of Baba Ramdev ji and Balkishan ji, we are getting the recognition at global level. So with the institution Patanjali Research Institute we have with us Dr. Asutosh Shukla ji. So, sir, namaskar.
Namaskar, sir.
For your gracious presence in this session. And we have Professor Kanchan Kohli in this session.
Yes, good.
Eminent
Good
academic,
name, ma'am.
eminent researcher in the field of pharmaceutical sciences. So certainly all of us will be benefited by her views on academia, by her views how we can get more and more in academia, how we can bridge the gap between academia and industry. So with our motivation, we will certainly be benefited. And numbers of others, like as Mr. Sunil Bhatt, as General Manager from Sanjeevani Parentals, Dehradun, and he is the person who is taking care of the export market of Patanjali Parentals in Russia. He is associated with this event. So, thank you, sir, for getting connected in this event. And one more colleague, Neeraj Sharma, is also here, and he is a person which is working as a team leader and with technical support in research and development department of Co-Science MedTech Company Limited, Beijing, China. And this personality is also with us in this event. So all the persons, all the technocrats from industry, good academicians from different institutions are with us. Dr. Amit Bhatia from Maharaja Ranjit Singh Punjab Technical University, Batinda. Shortly, he will also join this meeting. So, with these words, once again, I invite you on this session, sir. So, it's over to you, Dr. Prince, sir. And I would like to invite Hem Joshi, sir, for his deliberation, for his talk. Am I audible, Hem Joshi, sir?
Yeah. Thank you so much, sir.
Namaskar, sir.
Good afternoon, all. And I feel, very much happiness here to have an interview all means gurus in these universities and the teachers to interact with students who are the young managers and to have better interactions with the students. So thanks to all. Thank you so much. So I have rightly said, Dr. Vipin sir. So I worked with the professional organization for around 27 years. After completing 27 years in the professional phase, now I've decided to have my own entrepreneurship into the HR consultancy firm. So aim is to give the exposure, practical experience of the industries which is happening in the corporates and the manufacturing units, to share it with the students, so that to know about the. So please, this session will be beneficial to them and will get some practical knowledge from here. If any question is there, if they have any question in their minds, they can park it, they can note it. At the end of the sessions, they can raise the questions. We'll try to resolve or answer those questions. So next slide, sir. Next slide, please. Sir is there? Yes. Thank you. So this has been already explained by Dr. Vipin sir. Thank you again, Vipin sir. And I have worked in the different companies from automobiles to pharmaceuticals. In the pharmaceuticals, I worked for around 15 years as a HR head. So
This platform is mainly for the pharma students who have completed or who are going to complete the B Pharma and doing the M Pharma as well as certainly it will be useful to them since it will be more into the pharma sectors. So here Pfizer is a very big company in the pharma sectors. RFCL stands for the Remdesivir Fine Chemical. So this is the animal company of the Pfizer. This is the Indian company you know very well. Next slide, please. Yes. So the main objective is for the students, for the HR is that agility is the need of the hour. Agility, whether it's a physical agility or mental agility, both ways it needs alertness as on date. You know how it is critical for the HR, to identify the roles of the leaders and to focus the organizations as compared to last year's what it was decided and to propose for the future strength. So this is a very important, as you are aware that pandemic has created a disastrous ripples across the world, and all aspects of the human are jeopardized because of that. From the interactive sessions, which were used to be the classroom sessions in the Gurukul times, also in the Vedic traditional sessions, there were very rich gurus from Vasishtha to Chanakya, and the students were from Rama to Chandragupta. Ethically, morally, and societal point of view, they done good job to develop the country and develop moral values. So on that way, there is, where the role of HR is, the value system should be in strong way. Now, students naturally they are the young managers. Later or sooner they will apply for the job, will go into the professional fields. The professional fields is totally different as compared to the education fields. So means when they will join the organizations, they need many changes into them. Means one of the previous slide, please. Means these changes means adaptability. If we say adaptability in the sense of they need to adjust themselves with the organization's goal. Means what industry expects from the students, from the young managers, just to have those flavors into this. L&D, learning and development, learning, as you know, education, while grasping the more and more information, it's a part of the education. It's a synonymous of education, which is the information. So keep on learning and develop themselves physically and mentally. Again, the other expectation from the industry will be, if I give the example of the pharma students, B Pharma or M Pharma students, they will be having their mind, they want to be in a particular role. There are sections like quality assurance, in the production there will be, and quality controls. Definitely some of the key students, they might be thinking they will go for the quality. Some of them may be thinking they may go for the manufacturing or quality controls. So they should be open to, accept any kind of job in the studies. So there will be job rotations to get a certain positions in a particular period of time. And they should have the business acumen of the company, what exactly the business is into. For the pharmaceutical is the ever-growing business. It has a lot of opportunities in the market. And you might have seen in the pandemic season, as well as the private amount and get a good vaccines for human beings. So you people have good opportunities and good scope. If we compare the pharmaceutical industry compared to any other industries, they reward very efficiently, give a high level of increments, to pharmaceutical employees. And another change is that the expectation is that productivity. You know that any of the organizations generally run for the productivity. Means enhancing the productivity will be the key role, key expectation from the student. Other way around is that the communication skills. So without communicating the things, you will not understand what actually you want to be and where you want to be, right? So need to improve the communication skills through various learning modes. Means I mean to say we need to correlate the theory, means the book knowledge which actually you are learnings in the college and now working from home with the practical HR world. Why HR topic has been taken here, why I am emphasizing on the HR, since later or sooner you guys, means B Pharma guys or M Pharma people, maybe after some period of time, they will lead the department, they will be head of the departments, and sometimes maybe they will be factory managers or factory head. If they go into sales and marketing, they may be the sales head. So any of the head or the supervisor or the manager, he is equally 50% plays HR role. HR, human resources, is not only a role of a HR person. Means the functional manager is also a HR person. Why? He needs to take care of his employees who are reporting to him. Means the individual contributors, right? So that way means he generally, let's say for example, a HOD, if he is having the reportees of around 5 to 6 employees who are reporting to him, means day to day he need to coach them, he need to he need to mentor them, make the performance, he will interview them, right? He will need to take care of their leaves, performance management system, all the HR angles. So HR is important, human resources is important for each and every functional manager. Now, the another thing is that HR role leaders separately, it has a very fast future in the market. But the first phase for you people will be the HR. Means HR will collect the CVs from you, will you might have also done internship in between your second year, third year, fourth year, or maybe in the pharma guys, first and second year, maybe 2 months internship. Practically, you might have seen what the corporate or the manufacturing culture is. You might have happening, which is a totally different worlds as compared to the education world. Now, if you join any of the organizations, not necessarily you need to be expertise into one field. It really should be a T-shaped. T-shaped, I will give you a brief things about the T-shape. Now, the organization's expectations from the students or from you young managers is that you should know each and everything in the market. I have given the example of quality assurance. Even in the quality assurance, there are other sub-functional departments also, like one is the validation department, another is the documentation department, third one is the in-process quality assurance, there is a regulatory, statutory, all these departments are here who are filing the papers and those. So the person, the young manager should be open to grasp any of the role in the market. He should be open, not should stick to the any specific area. But later or sooner, if there is any job opportunities inside the organization is there, he can apply internally or can apply externally to get his long-term aspirations. So T-shaped professionalism is that means you should have generalists in 4 key areas, means data-driven. As you know, data is the most important as on date. If you have the data, data is the ammunition for you. If you have the data, you can interpret from the data and you can represent it before the management, where the management will get convinced based on your solid data, correct? And the business acumen, wherein the business acumen, you should have your work as related to the company's goals, company's purpose, vision, and objectives. It should be related to those things. And the other part is digital integration. In the digital integration, this is the digital world, how fast it's moving. with the gadget point of view, with the new technology point of view. So you should be techno savvy, right? And the other thing is the people advocate, which is the most important. People advocate. But people advocacy is taken here means people is the most important. If it's from the cross-functional department or whether it's your direct report is, means you need to get work done from the people. Mean it means that you should win the trust and confidence of the employees. There might be some of the as well as some external customers. Like, for example, like example of the quality assurance, they certainly need, some warehouse people who will supply the raw material to them. They need to interact with the manufacturing production people. So these will be the customer, and they need to interact with the HR admin departments to get facility management, right? And the QC person wherein the testing of the materials will be there. So people advocacy is the team building and the people relationship should be there. And one is in the red color, that's oblique functional competency. Apart from these general competencies which I have given to you four, one should be master into at least one competency. And one competency could be, in a given example of the quality assurance, like there were four sub-functions, means validation, documentations, IPQA, IPQC, and the audit part. At least one should be very perfect into one competency so that he can prove that, apart from other general competencies. In the next way, 6 key core competencies. In the next slide, I will more explain about the 6 key core competencies. What is that? The third point is health and wellness. This is the most desired chapter as on date. Means it's almost around 1 year, 3 months times, right? This because this pandemics, it has given a totally which is given the work from home, the mind, the mentality of the colleagues, of the employees who are working from the home, the people who are not interacting, there is no physical contact, there is no one classroom. So health and wellness is the most important things. My suggestion personally will be here as a HR point of view, since HR take care of the human capitals. So health and wellness in the point of safety is the most important. Means you need to perform yoga at least 15 minutes every day in the morning, maybe in the daytime, maybe in the late evenings, to get strong yourself, make yourself agile physically and mentally. Is the most important to maintain health and wellness, right? This is equally the need of the organization. If you are fit, the organization will be fit, and the organization will fit, and you will get the job. And if organization grows, it will give the money and the per capita income to the country will grow accordingly. The same way, upskilling, adaptability, skill, emotional intelligence. Emotional intelligence is the world. Right? We are right now we are doing the virtual interaction with the few peoples, but emotional intelligence or emotional may not work here. But necessarily, once there is the offline personal interactions, the emotional intelligence plays a big role. Sooner or later, the organizations are opening up with increasing some of the attendance. You might have seen in the news that some of the universities are also opening in near futures. So you will connect emotionally with them. It gives more efficiency to them. And cultural understandings is also key importance, means that there is a cultural changes while you go from your academic background to a professional background. The expectation of the teachers in the university is different, wherein the expectation of the the corporate world or the manufacturing unit or the professional organization, which is on the goals driven basis, works of the financials and non-financials on compliance part, the expectation totally different. So certainly there will be lot of expectations from you all guys. Another thing is artificial intelligence. Artificial intelligence is playing a big role as on date, wherein machine works a lot, like we are doing in the sense of the automations part. The Fifth one is the employee engagement. Employee engagement is the key engagement part in HR. I have already shared with you. You will be also a HR person. The Gallup survey, Gallup is the in the US, right? You might have heard. Gallup University say if employees are fully engaged, if the pupils are fully engaged means they are satisfied with their work. They are given the job opportunities, the environment, the bosses, the recognition, the performance management, the tools, the equipment to them. If they are engaged, automatically the productivity of that organization goes up without anything, by 18 to 22%. That's the result of the employee engagement, which is highly appreciable here, right? Another is the performance management system. And performance management system, it's a very long topic. I will not take much time. I think I have been given here time till 2:30, hope so. My timing is up to 2:30 or till what time is given, so that accordingly I will finish my chapter here.
Please continue, sir. Yes,
okay.
sir.
So performance management is the it has very vast scope in the professional world, in the professional organization. Sometimes there are quarterly performance management systems, yearly performance management systems, and annual performance management systems, wherein employee they are they get increment, and you should be happy as a pharma students and a pharma young manager that pharma companies, they generally give very high increment to those guys who are working for the pharmaceuticals. Even in the medicals, doctors, you might have seen, they are well rewarded. They are actively working, even if there is a pandemic situation. People are inside the home, but they are outside the world, into the factory. Regularly, they are working. So they are given in the 2008 recession also. This last year recession also, we had a per capita, the GDP has gone down. Even then, pharma people are there, really well rewarded. So there's the performance management system for the pharma people. Another is the recognition. Recognition is not necessarily a monetary. The recognition sometimes it could be like if you are interested with given example of the quality assurance department. Quality assurance department, apart from sub-functional department within their departments, they are interacting with their manufacturing department as well as IP quality controls. Quality control also has sub-functional department including microbiology and the HPLC systems and the quality control documentations and testing lab, etc. And means if somebody is doing good job for them, some of the managers, they are expecting some deliverables from theirs. They are giving them to well in time. You can at least recognize them by saying just thanks, right? Thanks to them, well done job to them, and appreciating them all the time. Should not, or the person, the young manager, any of the person, they should not discount themselves. They should not discount others. They should not discount their own activity. You have lot of opportunities, lot of skills, lot of stamina to perform, and you should recognize each and every things in the day-to-day world. Next slide, please. Yeah, HR capital. Why I have taken this as a HR capital? Capital words in the factory also. If you give the example, man, machine, and material. You might have heard these three words, right? HR, the human capital, means the people is the most important capital, irrespective of there is a building asset or these are raw material assets. They are taken on the top priority. HR is taken into the HR doesn't mean human resource department. It means any kind of human beings who is doing in whatever fields, from the security, from the housekeeping, from the quality, to a factory head, to a business head, to a owner. Means that HR capital is made to the organization. So it's given the value. Generally in the MNC culture, the respect for the people is counted more, and there are a number of surveys are taken into account. Means if the employee score is higher, employees are engaged more, the organizations are considered as an excellent and good working organizations in the corporate world. Now, in the HR, I have categorically taken it into three buckets. One is the inflow, another is the through flow, third one is the outflow. Inflow, wherein there is inflow, the peoples are coming in organization, the talent acquisition. Through flow, when they are into the organization, inside the organization, and certain roles are played by the different functions, different HODs in respect to the talent management, means the talent is the you all guys are the talent who are having the talent, right? So they are managed on the different tools, and outflow is the third category, means when employee leaves the organizations, it could be voluntary, it could be involuntary, right? Inflow is the recruitment and selection process. Recruitment and selection process is the key process wherein employees they are hired, they are scrutinized, they are gone through the interviews, they should know the interview techniques, how to present before the interviewer, you know. So this is the recruitment and selections. Different has different criteria for the recruitment and selection. So from my point of view, since I have worked in the MNC cultures and I've seen good companies, they had good, recruitment and selection procedures. I will get through in the next slide. Through flow means wherein there will be orientation, there will be learning and development for you guys. There is a specific training department inside the company who used to give you the soft skill trainings as well as the technical trainings. If I consider as a pharma student or pharma student, technical trainings means you will be learning more of your trainings, whether it's a quality assurance, quality controls, right? Or the manufacturing units in the production departments wherein you are into the injections, tablets, or the dry powder injections, so on, liquid orals, etc. Apart from this, technicals, in the two technical trainings means the organizations, they want to increase the productivity, or that they used to give trainings on the Lean, Six Sigma, 5S, DMAG, yellow belt is there, black belt is there. So many, operational excellence there, and number of good tools are available inside the company, inside the good company. You will get learned through those tools. You will be master into that, and you will have certain expertise. That is called the training and development. Because of these trainings, the productivity and efficiency of that organization goes up, and it becomes profitable in the world. Other is the succession planning. Succession planning is that if you are heading the department, definitely you need to develop your second line. If you will develop your second line, there is a successor for you. If you will develop that second line, means you will have the opportunity to have a job within the company or outside the company or across the company. You can apply for the seat. When you give the trainings to your second line, so that you can grow. Both way you can grow. PMS, performance management system, already explained. It gives the improvement chance to the employees. And promotions are there. Pharmaceutical company, they are well promoted, well rewarded, recognized, and they are given well monetary value inside the organization. And lateral career movement. It's a part of your job. And the third one is the outflow. Outflow sometimes is a voluntary and sometimes involuntary. Voluntary, you might have heard about the voluntary and involuntary. When the person resigns, he gets opportunity from outside the world, he resigns. Or sometimes there are the person who completes 58 years of age or 60 years of age as per the appointment contract terms, he gets the retirement. This is called the voluntary retirement. And another is the involuntary retirement. Involuntary retirement, which is the case of dismissals and redundancy in the people, if we are the people and there is a disciplinary issues, so into the involuntary system. The good organizations, they have the system, they give chance to the employees to improve themselves. They never immediately involuntarily retire the say terminate the employee. There are chances given to them. Next slide, please. Yeah. So this is HR, human resource management. I have given you around 10, 12 topics here. Why I have given 10, 12 topics here? As I have already said to you that later or sooner, you people will be also a HR manager since you will be having team. You will be taking in recruitment and selection of your people, will be taking care of performance management, will be taking care of learning development, success and planning, compensation and benefit means you will be designing their salary, right? Their salary, you will be having the budget for the salary, your departmental budgets, and there will be human resources information systems. Means HR systems wherein employee data will be there, wherein you will be take care of your employees regarding their birthday celebration, their marriage anniversary, their annual any other things, key data about their age, how much age is their qualification. You will record all those into their system, and you have also accessibility about their information system. HR data and analytics, wherein it will give the graphical representation you to present it before the management, how young team and how old team, how much experience team you are handling in company or in factory. HR compliance, it's a compliance of HR or it's compliant with the compliance of in the pharmaceutical field, maybe getting the FDA license, FDA, your documentation, your SOPs, so on. Workplace safety plays a most important role in any of the place inside the factory or outside the factory. It has certain rules, it has certain laws, it has certain labor codes also. Strategic HR. HR, I have given the chapter HR specifically, but you should also know, apart from the B Pharma technical thing, there is HR department also. Nowadays, HR is not HR, that kind of traditional or conventional HR, which used to be in the old times. It's a strategic HR now. It's a partnering with the business. Means on the day-to-day basis, it's equally partnering with the organization for making the profits with the teams, with the functional areas. Now separation, exit interviews and F&F. Exit interview is taken when there is a separation, when there is a voluntary or involuntary retirement, and F&F means full and finals, within their compensations, benefits, their salary part, their gratuity, their PF, which are the retirement benefit parts, right? Their leave encashment and their advances, their loans, all systems are taken into account, and the F&F is prepared. This is the key role of the human resource department. Next slide, please. Okay, now recruitment and selection. These are the most important things, recruitment and selection, which is part of the inflow, right? So I will keep focus into this, the inflow. So there is one system STAR. STAR is S stands for situation, T stands for task, A stands for action, R stands for results. A team who are taking your interview. If you are from the B Pharma or M Pharma line, if you are giving some interview, there might be panel of who will be from the pharma background, right? He will have the core skill of the pharma, so he will ask that from here the subject knowledge, whatever you have learned into your academic area. One person will be from the HR who will see your psychological and your education verification point of view. The third person may be there, will be heading the department, will be head of the functions, business head or the factory head, who will also take the interview. 3-4% teams will be there. They may all together can take the interview in a one board room, or they can take individually interview. So they generally ask questions from you on the STAR technology. Means they can give you a situations, means on that particular situations, even if you are a freshers, I will give you, since you are the young managers, you are the freshers, so I will not go as an experience point of view. I will use you as a fresher. I mean, what type of question they can ask. For example, if somebody has ordered very good marks in a particular subject. So what was the situations he got a good mark? What was the task given us to or he might have done some internship during his course durations of time. And what action he has taken to go through the situations and to complete that task. And what was the outcome? What was the result? Means whether he achieved the result or not achieved the result. Means this way the scenario is given based on this study mode. The judgment team, they analyze how efficient you are, how you respond to them. So you should be perfectly, should have proper study into you, right? And then came phase based on voice is clear. So I am continuing it. And what was the result? What percentage you achieved the result? Let's say I give one example of the salesperson. A salesperson going into the market, he's selling some product as a medical representative. Some of you people may be doing the job of a medical representative. He will be given certain tasks. He has to run the bike for let's say has the bike for 100 kilometers a day, 100 one side, 100 another side, 200 kilometers. Meaning the situation was maybe the rainy season.
Sir, you are muted.
Hello. Voice is clear?
Okay, sir. Okay.
So earlier also it was audible, right?
Yes, sir. Yes, sir. Yes,
sir. Whatever I have said, you hope you all have heard that thing. Clear?
It is clear. You may continue, sir.
Okay, thank you. Now CV. The CV should be good looking CV. And some of the companies, they generally give personal history form. Personal history forms wherein they have their set format. They say just fill up that format wherein you capture your personal things, your that I'll come into the next phase, how you will prepare that. An interview assessment sheet is there, wherein they mention the position you are interviewing, the name of the candidate, who are the interviewers, on what date you are there taking the interview, and estimated time. Generally, the estimated time, the reasonable, a good time is around 45 minutes time is calculated as a good time. Interview preparations, goal is to collect 3 stars. When I said the star, STAR, situation, task, action, and result. They generally give 3 scenarios to you, wherein they correlate and gives analyze. Based on that, they give grade to you. And interview opening times, they greet you, they will welcome you, right? They will explain about the position, they will explain about the work history form, they will explain about the job description, they will explain about the each and everything about the organization, and there will not be any confusion while you are facing the interview. And they will also answer you. If you have any question in your mind about the job descriptions, about the company roles and responsibilities, about the company agendas. And key background review is also there. Education is captured there, and your work history. If somebody is experienced, they can explain about their job 1 details, job 1, job 2, and job 3, whatever job he has done. Next slide, please. Yeah. So this is the planned behavioral questions. What types of questions on what basis are asked? This is the part of the star, wherein in the star, situations, task, action, and result basis. So these are the key behavioral leadership qualities. They want to see leadership qualities within you, whether you are capable to handle all those things or not. So means one is the delivering excellence, means the achievement orientations, taking accountability and quality of output, means what types of quality you have, means your result have. So they want to judge, they want to check, and how much accountable you are there to deliver your results. Another is entrepreneurial decision making. I mean, everyone who is, the interview candidate, he should have an entrepreneurial mindset, ownership mindset. Means he is honoring the things. Means independently in the current roles or functional know how, entrepreneurial mindset, and the long-term thinking. They want to check whether he has a long-term thinking or not. Means the person who is giving the interview, that he should not leave the types of 1 year's time. What is his future goal that they want to judge, right? And business orientations mean understandings on role, what the role and its impact on the business, functional know-how, enterprise mindset. Enterprise mindset means whether he knows about the company or not, where he is going for the interview. For example, by going for the interview in Kedala, let's say for any other organizations, ABC organization, he should be well prepared with the, the product, about the number of employees stamped, about the details of the company, goals and features, right? So he should be well prepared. And the long-term thinking. The fourth one, which is the team leadership. So the person should demonstrate the team leadership, whether he is holding people accountable or not. Means he has the capability. Even if, in the college times also, you are the students, you might be doing some of the voluntary works outside the college premises or maybe outside your home also, right? So do you have the team leadership in classroom, in the factories, from other departments, and whether you are recognizing your people or not recognizing, means you are appreciating them or not appreciating, so that they will identify. Building people's capability means recognize and acts on capability needs. Empowering teams or people means whether you are empowering your people or not empowering. Means the people who are reporting to you, how you are empowering them, giving some additional tasks, giving some different tasks, right? Not the routine task, apart from some other challenges. The n number of audits day-to-day happens into the pharmaceuticals manufacturing units. So whether each and every person are participating into that or not, whether each and every person are part of the continuous improvement and operational excellence or not. This is where the accountability is there, right? And these are the providing opportunities, which is part of the coaching and mentoring as well as whether you are coaching them, giving them mentoring or not mentoring. Working across boundaries means how, apart from there is, in the organization, in the professional organization, when you join the organization, job description is already given to you. Means this is the job A, B, C, D you need to perform. After 3 months of time, you are analyzed by your supervisor, your HOD, means that job descriptions he has prepared, whether you are performing well or not. There will be mutual dialogue between you and between the manager, meaning you are delivering those things or not. He will clearly communicate to you, okay, out of this 10 job, you have learned A, B, C, D, 7 to 8 items. Still, you need to learn 3 more things, and you have so and so another 3 months of time. So there is a good communication in between you and your managers, right? So that is the understanding. And in the working across boundaries, if it's a good team players, impact and influence, understandings, others, collaboration and integration. This also play a very active role while identifying you as a candidate. After scoring a good score, while doing these competency-based mappings, if it's a motivational fit, the final comments are given and the candidates are selected and they are well communicated, they are placed as per the compensation structures defined in the as part of the HR policy or company policy. Right? Next slide, please. This is the CV. So, curriculum item. It should be very simple. Generally, resume or CV, it's almost same thing. Resume is a smaller one, CV is a large part of the resume, right? But the resume or the CV, curriculum vitae, the fonts and colors should be very simple. To max, it should have 2 pages, not more than 2 pages. If it's coming within 1 page, sufficient. The maximum information should be captured within that CV. 2 pages sufficient, should not go more than 2 pages. Initially, you should capture your name, contact details, means number, and your email ID, etc. on the top of the resume, wherein if they need to connect with you in the future about the result of the interview, they should immediately locate your name on the resume. And professional profiles, if you are a professional, you have some experience, you have some internship, it should be introductory. It should explain about your abilities and aim, your skill, experience, and your knowledge. That professional profile, brief professional profile should be there. It should be, clearly visible to panel members. Do not forget to capture your core skills. Correct? Like I explained to you about the quality assurance department. For the quality assurance department, certainly they need certain core skills. But if you are applying that for the particular job, quality assurance, not for the quality control, but you should have the core skill clearly explained and detailed in the experience. Orders, right? You should have your key responsibilities, what you have handled, and key achievements, what you have achieved during your tenure. Education and qualification, most important part, you need to mention and qualifications in the resume. References, it can be if need based, not necessarily need to put the references at the right times, but on the need based, you can provide the references. Since sometimes the references very good role while you are appearing for the interview to any organization. Hobbies, do not forget to write your hobbies and interests, your interest area. If you have done some voluntary work, which is for the social cause, for the society, and for a good cause, do mention that part. It will count a lot while giving interview. Sometimes the panel member may ask from your hobbies and interests also, wherein you've done some voluntary work for the society. They can take some questions from there, wherein on start basis, you can explain how you achieved that result in a particular situation. Maybe there's a situation of this pandemic in the last year, how it has, disaster, how it affected the life of the human beings. You can explain all these things too. And finally, the commitment that all above whatever you have mentioned in the CV, that's true and that's correct. You can sign it CV and put your name on that. Yeah, this is all from my side. Hope you understood it and it might have given some clarity to you all young managers.
Thank you very much, sir, for your nice deliberation. And we are surely hopeful that each and every young manager will surely practice this knowledge in their fields, in their interviews. Thank you for your deliberation, sir.
Thank you. Thank you so much. Thanks to all.
आपने समय निकालकर हमारे लिए यहाँ पर आए. Thank you very much, sir. Now, in continuation, I would like to invite Mr. Neeraj Sharma, who is team leader at Co-Science MedTech Company Limited from Beijing, China. if he is listening me.
Yeah. Yes. Hello.
Hello.
Yes.
Yes, sir. Welcome, sir, to this
Okay.
virtual platform of Ashdi Padap MahaKumbh, celebrating National.
Just wait. I just.
I request all the participants to please mute themselves. Wait for a while, sir. I'm making you a presenter.
Okay.
The question and answer session will be taken at last of this academia industry meet. So I would like to request all the eminent speakers to kindly remain connected to the session. So welcome you, sir. Neeraj, sir, please continue with the deliberation.
Okay.
ये सर जी अब आप बताओ इसमें करवाना है कि नहीं जाकर।
So you guys are able to see my screen?
Yes, sir. Please continue.
Okay. So good afternoon, everybody. And I think some teachers are there, so I will use respected sirs, and some students are there. So good afternoon, every students, every guys which connected to this unique platform of Gurukul Kangdi University, and this is a good like collaboration for academic and industry. So in this presentation, I try to explain how we can develop the product for US market, or you can say regulatory market, because most of the companies or industries are working for the US market for the development of product. So I just try to explain there how we can develop the product, what is the procedure for the development. So in my presentation there is a background, development steps, excipients information, and excipients uses. So because we all know India is a like all around the world top 5 generic development country. India have good developer with respect to the RLD, reference listed drug. We are developing generic product and file ANDA, abbreviated new drug application. This is the like good logic of the Indian companies to go for the US market, and US government also promote these things because India developed low cost product with respect to the innovator and have good bioequivalency, good effectiveness, and good quality. So this is the quality of India. So now India have good or brand of the ANDA filing, like Dr. Reddy's, Lupin, and previously Ranbaxy. Ranbaxy is the like a huge platform for the ANDA filing in the US market. So now we start for the steps. So there is like 21 steps how we can develop the solid dosage form. So solid dosage form like tablet, capsule, and some sachets. These are the solid dosage form. So first of all, when we start the product, so we search out all the information of reference listed drug, or you can say innovator, from the FDA site, Orange Book, or other Google or other search engines. Then we will purchasing or sourcing the active, means the API, like because we when we purchase some tablet like paracetamol, so these are the active. So as per the like regulatory policy or the US requirement, we approve 2 sources for the API. Then we evaluate the active substance. After that, for active and excipient purchasing, and then active testing. So these are the initial steps when we choose the innovator and parallelly select the active substance source, because for the US market we choose or we select API which have DMF, drug master file. Without drug master file we can't use API for the US or regulatory market development. So this is the important thing for the API selection. For next, like innovator product purchasing. So innovator product purchasing, we purchase or we import from the US or the market which you want to file the dossier. You purchase innovator from that country. So for purchasing, have some formalities to justify the government how to utilize the product or why you purchase this product. So this is the innovator purchasing. When the innovator came in our site like R&D or the plant, then we will evaluate this product like physically and chemically. After that, we start our development part. I mentioned prototype initiation, or you can say initial development trials. So in this prototype initiation, process selection, excipient selection, like process selection, which process we need to do, like wet granulation, or you can say dry granulation, or you can say direct compression, or some other like top spray, bottom spray. So these are the different procedure. Wet granulation I just explained because some students are also there. So quickly I explain the procedures, some small things. In dry granulation, we will use roller compactor, and in direct compression, we just blend the API and excipients and compress the tablet. And in wet granulation, we will granulate like API and excipients in RMG, rapid mixer granulator. And in top spray, there we will perform, there is a spraying in a fluid bed processor, and bottom spray in same equipment, but the mechanism is different. In top spray, the solution came from the upper side, but in bottom spray, solution spray from the bottom of the machine. So these are the different kind of manufacturing approach. This will totally depend on the molecule, API. If molecule is sensitive to water, so we can't use granulation with water. Or if molecule is sensitive to exposure like oxidation, so we just blend and then compress the tablet. So these are the process selection, and same thing is the excipients. So we when we develop for the generic product, so we are trying to use same excipient like innovator. So these are the approach. Same time we draft our project plan, like some how we can drive the development and define QTPP and CQA, so that we can capture all the things accordingly. Side by side, we develop analytical method because one more thing I just explained, in industry everything is in calculative manner. We have limited time to develop the project because every formulator have like limited time. On that time we need to do this project. So in that manner we will perform or we will plan the project. When our prototype finalize, after that we will perform API and excipients compatibility. So in API and excipient compatibility, we mix like API and different excipients in proportionate manner. Like if in our dosage form, API is 50% and some diluent like some excipient is 50%. So we just mix 1:1 ratio and put into the glass wire and put into the stability chambers for 1 month. After 1 month, we analyze the potency, assay, and impurity of the API and excipients. If all things are okay, means this combination is compatible to each other. So this is called API and excipient compatibility study. So this after that we will start formulation and process study. So in this study I mentioned there is a OFAT, one factor at a time, and oblique DOE. This is a software for the design of experiment. So when we put some values in this software, when we put some values in software, they design the experiment, like how much experiment we can do for this product, and one is offered one factor at a time. Suppose we are using 20 mg excipient, so we can study if this excipient will decrease or increase in some proportionate manner, what will be the impact on the formulation. So this kind of study is performed during formulation and process. This is the most important phase or most important part of the development study. After that, we will, when our formulation, all things are finalized, we will go for the lab batches, stability lab batches. So we design the stability batches and then pack same as the innovator container or closer, then put into the stability different condition like 40-75, 30-65, 25-60, and 30-75 accordingly. But mostly we will perform accelerated condition 40-75 for 6 months, and we consider this data 3 months exclusively. Then we will start for the next step. So this is the development phase. Down after that we will project plan update and risk update accordingly, which we have performed from initial to the stability batches. Then data compilation, review, all these things. So this is the development phase. After that we will plan for the PO batch. This is process optimization batch, because in R&D we just perform in 500 gram or 1 kg batch. Then in PO, process optimization, we will go for the plant. Then we will perform 100 kg, 80 kg batches. Then optimize the process. What is the impact when we increase the batch size? Because there is a big machines, all things are changed. So this last OFAT or DOE trials, this will be helpful for here, because we get some space how much changes we will do during the scale up. After scale up, we know when we enlarge the batch or enlarge the product, what is the impact. And if PO batch is okay, all analytical activity of PO batches will okay, then we will go for the scale-up batch. So in scale-up batch, we have performed exclusively for the analytical things. Just wait.
Hello, Neeraj sir.
Yes.
You are not audible.
Okay. Hello. Hello.
Hello.
Hello.
Please continue, sir.
Okay. Okay. So before these slides, my voice is clear, is audible.
Yes, sir, audible.
Okay. Okay. So at the end, from scale up batch, we will go for the exhibit batch or pivotal batch. So this is the main part of the final part for the development to scale up. So in this exhibit batch or pivotal batch, QA collects some sample for the bio study and send to the bio center, and they perform bio study. And before ANDA submission, we review all the raw data of development or LNBs, bio study report, evaluation, all the interim report, and make a document. This is called like dossier, and ANDA submit to the FDA regulatory body, and then they review this document. Sometimes there is RTR, refuse to receive. They refuse after the receiving, and then sometimes they accept. If because in FDA have some stringent like procedure for all the things, like documentation also. If your ANDA is not in proper format or like proper numbering, so they just RTR, refuse to receive. uh, Otherwise they accept, and the process is going on like acceptance, review, some queries raised by the FDA, then company will correspondent to FDA, and then FDA is satisfied, then FDA will come to the plant site, then review all the facility or the product related document, and give the approval of the product. So these are the total ANDA submission steps, because this US FDA have some stringent guideline. From Europe market also have, but most of the time US is tighter than the other market. So in this phase, all the companies try to maintain quality wise all the things with respect to the FDA guidelines. So these are the things I just explained to you. And next one is like some excipients, because we all know, suppose we purchase some tablet like paracetamol, metformin. So everybody know this is a metformin tablet, but apart from the metformin there is a some excipients. These are the carrier to carry the API in a dosage form. So these are equally important like API, because without excipient we can't formulate any dosage form. So these are very important, and I just try to explain some small or general categorize of excipient like diluent, binder, disintegrant, glidant, anti-adherent, lubricant, film forming agent, control release polymer for sustained release dosage form, entry coating polymers, solubilizer, dissolution enhancer. So diluent is the most common or most important part of the dosage form. For example, like MCC, microcrystalline cellulose. These are the like diluent, lactose, mannitol. These are the such kind of diluents, and we will use these as a carrier of the API and binder, because in wet granulation we formulate a granules, so we need some excipient to adhere all the powder as a like good granules like starch. And in previously we are used gelatin, but nowadays gelatin is not too much used because have micro problem. So starch, povidone, these are the binders we will use in the dosage form. And for the disintegrant, because every formulation when we swallow, so the tablet disintegrate in our stomach or sometimes our intestine. So disintegrant is equally important. So like cross povidone, cross carmylose sodium, sodium starch glycolate, these are the super disintegrant and we use in our formulation. And the third is the glidant, like colloidal silicon dioxide. These are the glidant we add in the formulation. Anti-adherent. So talc is a anti-adherent because when we formulate granules, then compress in the compression machine, so sometimes there is some sticking, tablet stick in the punch. So this kind of thing we need to avoid. So we add anti-adherent and lubricant. For example, magnesium stearate, calcium stearate, stearic acid. So these are the lubricant. And at the end, like film coating, because our tablets, we know our tablets red, blue, yellow. So we add some film coating agent. So when I started my career, so that time we prepared in house, like add some HPMC and some plasticizer, PG, and then solvents. So these in house preparation. But as time is running, Oppa Dry is more commonly used from like Colorcon and other companies. So there is a ready mix Oppa Dry, have the like some coating agent like polymer and some coloring agent and some plasticizer. And sometimes this like coating formulation is customized as per the formulation or the scientist. So these are the coating agent. The 7 is the control release polymer. So because we all know some tablets like sustained release and extended release. So that time we used some control release polymer like hypermillose, HPMC, and some Udrazit, different grades. So these are the control release polymer. In HPMC have different grade as per the viscosity, so that they adhere or they control the release of API for particular time, like 12 hours or 24 hours, like this. So we add these control release polymer in the dosage form. And then at entry coating polymer, we when like aspirin, to avoid the degradation of in stomach of the product, then we use or we coat the tablet with entry coating polymer. And to degradation of acid, so we use Udrazit, ethyl cellulose, these kind of polymers. And the 9th is the solubilizer or dissolution enhancer, because API have different class, like BCS class category wise. So sometimes dissolution has some problem. So that time we use some solubilizer like PG 6000, Oloxamer, different grades we have used to increase the solubility or increase the dissolution. So these are small explain on the excipients chapter, and there is a this one is the key book for all the formulators in industry. This is a handbook of excipients. In this book, if you want to know like which excipient we need to use in how much concentration, and which excipient have particular which category, so these kind of information you can take in handbook of excipients. This is very good book for the industry perspective. And when sometimes in colleges some projects is governed, so this book I think is helpful for the guys. Yeah, this one is the inactive ingredient search for the approved drug product. So because in regulatory market, when we develop some product, so particular amount of excipients we need to add. So when we add some MCC or some sodium lauryl sulfate, so in this site we need to add the excipient name, then you get how much maximum amount you can add in one tablet. So these kind of information you get, so that because if suppose you prepare the tablet and you have no idea how much is the maximum concentration, and you prepare the dosage form, so at the end some regulatory body reviewed this thing and they are saying this is wrong, so that time you have no option how you can justify the things. So before you go for the next step, then you review and you get the idea of the excipients concentration, how much you add in one tablet. So these are the small things which I explained. If somebody have question, so you can ask. Hello. Hello.
Hello. Yes, sir. Hello.
Yes.
Hanji, sir.
So it's completed. So
Okay. Thank you very much, sir.
Okay.
It was really nice presentation by Mr. Neeraj Sharma from Beijing, China. Now I'd like to request our eminent speaker, Professor Kanchan Kohli from Jamia Hamdard University, New Delhi, for her deliberation. You are welcome, ma'am, to this virtual platform of Ashdiya Padak MahaKumbh organized by Gurukul Ganga Vishwavidyalaya, Haridwar. Ma'am, please make yourself unmute. Ma'am, please unmute yourself. 1 second. Wait for a second. Yes, ma'am. Kindly unmute yourself.
Yeah. Because earlier, but it was not. Yes, some problem. Right. So am I audible now?
Yes, ma'am. Yes.
So, all is coming. I don't know. So first of all, I thank organizer, Honorable Vice Chancellor to organize this type of a program, and this type and the whole of the month are busy organizing, and I know it's very difficult to organize. I mean, at international particularly, and I'm finding daily one or other some program is there. So I'm thankful for organizer and me regarding with this industry institute action, and I really want to discuss why do we need interaction? Am I audibly or some
Your
echo
voice
problem
is
is going?
echoing. Ma'am, I think two windows are open. Your voice is echoing.
That's that my wife. I don't know why.
You
Is it
can
okay?
mute.
Is it okay now?
Please
Hello.
continue.
So why do we need industry interaction as the topic is there? So we already there is all known as pharmacy of work, and particularly during COVID time, seen that what's the importance
अब तो हो गया ठीक
of pharmaceutical?
हिसाब में डाल देता हूँ फिर भी खरीद के लाया क्या
Hello. So we know India is already known pharmacy of world. Earlier also, COVID time has come up, our pharmaceutical industry has come up like anything in the world, and a lot of news or generated lots of time, finding that many of the products been exported during COVID time, they are in export. So what we need is government has recently announced PLI, that is production linked incentive for pharmaceutical industry, and lots of funds are there, and because of the industry interaction, we can use those funds in a better way, and definitely if not new molecules, we can change the delivery systems, in which particularly we are bothering about the bioavailability of the drugs. So the industry problem is here, that how to the bioavailability of the class 2 to class 4 drugs, in which the bio problem is there, and about % medicaments are there are having solubility and bioavailability problems. So by the interaction of the institute and the academia, industry and academia, so industry will can tell to academia or all to academic people how they can go ahead solve this problem, and even sometimes the problem of problem of excipient is there, so those can be provided by the industry to help academy, and without the help of the industry, the academic people cannot help. So industry institute is very important, and institute prepares and provides skilled personnel for industry, and industry produce medicine for mass. The interaction enables the institute to develop ever-changing requirement of industry. The day is not far the India shall be manufacturer of medicine and provider of skilled manpower. So how the manpower provided? As I was listening before this Mr. talk, HR talk, in which please don't change. First slide, first slide, first slide, first slide. After this, Next, please, next slide. Yes. So, why do industry institute interaction, increasing complexity of research and business environment? The industry academia here is all about knowledge transfer, experience, technology transfer. So as we need, they, I mean, IPR, filing for IPR, definitely we need MOU note to it, the interaction with the industry, and without the help of the industry, simply it is not possible. Suppose we have generated product at research level, so definitely unless it is not scaled up, the pilots not being done, that's no value. So for going for the pilot studies, because all of us know that institutes don't have much infrastructure in which we can do the pilot studies. First is funding problem, and the second is the infrastructure problem. We do need the help of the industry. So where the industry needs us, I mean, giving the latest information and all the literature survey, so definitely the industry help. Industrial organizations, money and workforce. Academics, what will be giving the extensive knowledge, experts in their active. Therefore, the two are dependent and need to work in synergy. If synergy is not there, then our knowledge go in waste. So whatever the academics research being done, they will go in waste. Of course, for industrial point of view, they are using our research papers, latest papers, and using those papers, publications, definitely we are helping to industry. And newer research is only going in academic only. No industry carrying out. And by publications, by filing IPR, definitely industry help. It's true. The two are mutually dependent. Next slide, please. Next slide, please. Next slide, please. Next slide, please. So, win-win partner. So, in win partnership for industry, academics knowledge based to improve the industry cost, quality, and global competition, updating and upgrading the knowledge, access to extended networks, thinking outside the box, find new talent, hire, facilitates research discoveries, reaching the people who need them and an affordable prices. Combined credibility increase. Discoveries from the academic institutions to the market. And this was for the industry, where the industries will be benefited. And how the academic be benefited, they need the deepening perspective of teachers and researchers, secure placements, improved curricula, hands-on training, help students development vocational self-concept, acquire job relevant skills, provide informed career decision making ability, career opportunities, awareness of the industry trend. So, looking on these industry and academic institute, we have seen how the industry and academia will work from hand to hand. And for the IPRs, as we are finding US patents, European patents, so definitely India will also come up in increasing the number of the IPRs, and once the IPRs will be increased in number, definitely academics people will be helped also, because they will be also having their lives, and industries can also earn better way, because they can go for the latest formulation development, standardization, optimization, and calibration of the product. So since we are at Gurukul Vishwavidyalaya, Kangdi Vishwavidyalaya, we are more bothered about the traditional collaboration. So we are more bothered about the what the traditional herbs were there. So far they been not patented. So how we can develop traditional medicines into the modern drug delivery system? And as we all know from the pharmaceutical background, that in our herbal products or the traditional products, bioavailability is the main problem, solubility is the main problem, and we don't know about the distribution of the drugs also. And all these factors, we go collectively, lot of the research potential is there. Even if we touch even the masalas kitchen and we start working on, as we are finding kalonji, for example. So kalonji is new to us. We are using from the long time the kalonji pickles. When our mother was preparing the pickles, they were using to add the kalonji in it and we've been eating. But today, because of the herbal knowledge, think that how the kalonji can be explored, so we can have the phytoconstituent, that is the thymo, and the thymosin is having an anti-cancer activity. Even we explore for the anti-cancer activity, so we can go ahead bird and can file the IPRs, which to the extent that we replace this synthetic drug from the market, and that time is not far where we can replace many of our modern drugs with the traditional one. We can bring the traditional drugs in, and I think Gurukul Kangdi Vidyalaya, they are having all this awareness, and I'm thinking Dr. Satinder Rajput is there, Professor Rajput, who has recently joined the institute, and I know his caliber. He is very good in formulation point of view, that pharmacological point of the drugs he can carry well. And he can very well handle the animals, so that the preclinical studies of the many herbal formulations can be done. And I think going to explore very soon, and wherever our help is needed as an academy, so we are always there to share our knowledge. And here I'm finding too much is there. We can go for the industry institute interaction for the various studies, because we know one teacher is very good from the formulation point of view, other is very good from the herbal point, and another good from the pharmacological points of view. So why not we can work combinedly, and we give our country some modern products that can be exported also. So in way we can go for the promotion. So how we go for the promotion of the subject? By organizing workshops, which you are doing very well from the last 1 month. You are busy in organizing the workshop, conference, and symposia, and I'm regularly watching, regularly attending some of it also. Organizing workshops, conferences, symposia with joint participation of the faculty and the industry. The example is very well today's example only. Encouraging industry professionals to
institutions to deliver lectures, and I think pandemic time, during long time, this point that's can bring international speaker, and that's what should be doing it. So experts from industry help in curriculum development of staff in various industries, professional consultancy by the faculty to industry, joint research programs, joint guidance of the faculty and experts from industry to students in project work, R&D laboratories sponsored by industries at the institute, scholars fellowship instituted by industry at the for students, practical training which the students go in various industries when they are giving the
offline right to invite. Hello, ma'am.
Practical students in industry, enhancing the importance of applied research. So I think time is short, so we can elaborate each of the point and we can look forward for the industry institute interactions. So with these remarks, I'm really thankful to organizers, participants for a good listening, and thanks to all.
अब सारी चीजें ही लाके लगती हैं
बाद उठाते हैं। हेलो कंचन कोली मैम। I think we are running short of time. I will request Kanchan Kohli ma'am to conclude, and I will also request her to visit the campus offline because we want to listen you more, your caliber, your research, your academic experiences that will help us. So I request Kanchan Kohli ma'am for the concluding the session so that we can go for the another session.
I've completed, and in conclusion, I like to say that we want the institute partners so that we can go ahead and our country can progress. Thank you so much.
Thank you, ma'am. So the experts are today visiting offline tomorrow, so we'll listen more tomorrow to the Shukla sir from Patanjali and Sunil Bhattaji. Now I will request the moderator for the other session. So moderator Dr. Poonam Negi, if she is online, she can take over the session. And I will also request Dr. Brahmeshwar Mishra ji and speaker of this particular session, N.V. Satish Madhav. So I will request open your video so that we can coordinate further. I'll also request Abhishek Bansalji, coordinator for the session, kindly join so that we can have the another important session.
Hello. To Poonam ma'am.
Yes, I'm there.
Okay, ma'am. Ma'am, we are waiting for our chairman, sir, for this session.
Okay,
So
okay.
as soon as he will arrive, we will
Yeah,
continue.
yeah.
Hello. Our chairman sir of today's session is connected with us.
Okay.
If my voice is audible to him, kindly reply, sir. I am audible to you. Professor Mishra, sir. If this is clear, please reply. I think he is facing some technical problem. So we are waiting for him to join. Welcome to our chairman, sir, Professor B. Mishra, sir. If I'm audible to you,
I'm audible. I'm audible. I'm audible.
Yes, yes, sir. Yes, your voice is clear.
Okay. Okay.
So welcome, sir. Now I would like to request our moderator of the session, Poonam Negi, ma'am. Please continue with the session.
Yeah. Thank you, Dr. Abhishek. So I would like to welcome you all to this wonderful session wherein we have with us Professor B. Mishra, who will be the chairperson of this session. And we also have our very illuminary scientist, Professor Dr. N.V. Satish Madhav, who is the speaker for this particular session. So before I proceed further, I would like to invite Professor B. Mishra, who has a very illuminary career in pharmacy. He is currently working as Professor of Pharmaceutics at the Department of Pharmaceutical Engineering and Technology, IIT BHU. He has served as head for 4 years in the same department. He has 36 years of teaching and research experience. He's reputed academician and acclaimed researcher. His area of research is development, evaluation of novel drug delivery systems, including nanoformulations. He has guided 2 postdoctoral fellows, 26 PhDs, and 74 MPharm students. And he has also published several journal articles, 15 book chapters, 2 books, and 3 patents, which are all published. Till date, he has an 6254 citation, having H index of 39 and I-10 index of 122 to his credit. He has been the leadership, he has got the leadership for academician program LEAP sponsored by Government of India, MHRD, in 2020. He is an acclaimed researcher, which is evidenced by the name appearing in top 2% of the world's scientist list in pharmacy pharmacology category, recently declared by Stanford University, USA. So we are so lucky to have you. And I would like to invite you to start the session and say a few words about this session. Thank you, sir.
Thank you, Dr. Negi. Thank you, Dr. Negi, for a nice introduction. It's my proud privilege to chair a scientific session of which is going to be delivered by my own friend, very good friend, Dr. Satish Madhavji. And as you all know that he has taken a great venture in his life by changing from academics to industry. And he was a principal for so many years at DIT University, Dehradun. And then he has been doing very good work there in terms of academics as well as research. I know personally because I have evaluated some of the PhD theses of his students. And now he changed to industry. It's good. I think for anybody, it's always good to have a change in the life. And he shifted from academics to industry and joined a pharmaceutical R&D lab of a very reputed pharmaceutical company in Hyderabad. So I welcome as a chairman Dr. Satish Madhav to this scientific session. Gurukul Kangdi, though I have been attending very first time, Gurukul Kangdi Haridwar, any program, either though it is being online, but I have not even visited physically also till now. It will be my pleasure always to see that if I can find some time or some occasion or some opportunity, I would like to personally go to Gurukul Kangdi and see the facilities and other things which are going on in terms of academics and research. So they are doing good work. They have arranged this organized this conference. Dr. Abhishek Bansal I thank him that he is in contact with me and he invited me to chair this session, which I readily agreed because once I saw that my friend Dr. Satish is speaking and Poonam Nengi is moderator. So it's my always privilege and pleasure to chair this session. And I think my best wishes are there with the organizing team, especially Dr. Bansal. And I hope that all the participants till now must have acquired a huge amount of knowledge in the field, in the topic of the conference which has been going on. Without wasting further time, I think I do not know that whether the chairman has to be introduced by whom, because I do not have any biodata of him.
Yeah, sir.
So
no worries, sir. I will introduce him.
Okay. So I would request Negi madam to kindly introduce the chairman, and then I would request Dr. Satish to kindly proceed with his lecture.
Yes, sir. So we have with us Dr. Satish, who is a PhD from Jadavpur University in pharmaceutics, MPharm from Bangalore University. He has served as a principal and professor, HOD, Department of Pharmaceutics in Noori College of Pharmacy. He has also served as principal and professor, HOD, Department of Pharmaceutics in Pharmacy College, Ituara Chandreshwar, Azamgarh. He has served as a principal and professor, HOD Department Pharmaceutics in DIT University, Faculty of Pharmacy, Mussoorie. He's currently working as a head in formulation research development in Vital Therapeutics and Formulations Private Limited. He is having 25 years of experience in administration, teaching, and research. He has co-guided 23 PhDs and 56 MPharms, which is a very, very huge number. He is a pioneer in exploring potentially of oro-soft palatable drug delivery platforms. He has isolated more than 130 biopolymers from natural edible sources. He has explored the potentially of various drug delivery platforms like eye to brain, ear to brain, translingual drug delivery, transnavi drug delivery, transvermilion drug delivery, and cranial drug delivery, and various bio-nano formulations. So with this background, now I would like to invite Dr. Satish to start his presentation and his very, very interesting topic, which I'm really interested in. So over to you.
Thank you, madam, for your kind words. Thank you, sir, for your kind words. Now I'm going to start the session. Today, I chosen a topic on smart, novel, nature-derived biopolymer functional agents for delivery of therapeutic moiety via trans-Navi route. Why, how it has been evolved the now Navi. This was initiated way back had given a term called as a trans-naval. Later on, to a thing called as trans-nav delivery. Nabhi means is a naval in Sanskrit. Trans means it is moving the molecule from one end to other. That is Nabi and to systemic circulation. Here I'll be giving some novel polymeric agents which are derived from the nature and which can be used for delivering the molecules, especially therapeutic molecules, via trans-Nabi. Next slide, please. Next slide. Next slide, please.
Wait a second, sir.
Just I'll brief about the Nabi. Nabi clinically known as umbilicus. Nabi is clinically known as a umbilicus, umbilical button, and umbilical dip, navel, tummy button. It is a median depression. It indicates the site of attachment of umbilical cord before birth. Here, during the development of the baby, all the nutrients which are being obtained from the mother to the placenta, it will be to the baby through umbilical cord. After the birth, this cord will be placenta. The Nabi exactly lies in level the junction between the L3 and L4 quadriplegia. Next slide, please. Coming to in perception of Ayurveda, Nabi is called as a DNA of our soul and the best source of transmission, transformation, and healing. And Nabi is considered as a vital area in which arteries, tendons, flesh, veins, bones, joints meet, form in a location of a life. And this marma exactly containing around 72,000 of nadis, nerve endings, as well as a blood vessels. And that's why this is the thinnermost area of the abdomen where the skin is very thinner in nature and our body is consciousness is located in the navel region. Next slide. And when you see the anatomy of this Nabi, the umbilicus is a depression, the left bottom of which is concealed by psychiatrists, left by throwing off the cord. The psychiatrist is drawn inward by the retraction of the umbilical vessel and the special tissue which has been surrounded by. This is the umbilical psychiatrics which has been formed after the detachment of umbilical cord from the placenta. Next slide. When you in the abdominal area, the thinnest, it is the abdominal wall containing several layers of the skin, layers. One is skin, superficial, and superficial sheath of the abdominal muscle, rectus abdominis, deep layer of the sheath, subperitoneal connective tissue, and peritoneum. All this abdominal wall are fused at the umbilicus. This umbilicus or Nabi is considered as a center point. Then from this center point, all the blood vessels will be originated and it has been helped in formation of various parts or organs of the body. Next. The umbilical is a depression been covered by the squamous epithelium. Apart from that, it is a democratic issue, and it is being considered as the most innermost area of the abdominal wall. Since it is the most innermost area of the abdominal wall, and this is the main door for entry of a nutrients to the fetus from the placenta, as I told in earlier. But now, after the formation of, after the birth of the baby, this abdominal wall may be closed with a thin layer. So till today, in the therapeutic delivery, there are minimum number of research has been focused, but way back 12 years back, we are focused to deliver the therapeutic moiety. In Ayurveda, there is treatment of local as well as a systemic via Nabi therapy for chapped lips for indigestion purpose. By pouring the oil into the Nabi, they used to treat various the gastric problems and other problems related uterus problems. They are going to treat by using Nabi. Next slide. Next slide, please. When you see the blood supply, during the development, the fetus will be receiving the blood from the placenta. One is with a one artery and two veins, means maximum blood, purified blood will be received with one artery, and the impure blood will be circulated to the placenta. That is with the help of a two veins. That is umbilical vein, which passes to the liver along with the sensory ligament, umbilical artery passing downwards, outward to the bladder region. The uretrus passes to the bladder region. This is the location the nutrients move inside and outside of the next slide. Next slide. Next slide. Umbilicus received the flow by means of three distinct deep sources. One is right and left deep inferior epigastric. Ligamentum teres hepaticum, which is going to circulate to the liver, and the medial umbilical ligament. These are the three arteries which are going to originate from the umbilicus, and a few para-umbilical veins which connects to the portal vein, through which the umbilicus with the ligamentous teres. So because maximum arteries which has been veins are reaching to the liver, and we explored a method for delivering the API molecule to the liver targeting, whereas atorvastatin and we had tried, and we got in a very liver to the site specific, especially in the liver region, via transnavi. Next slide. Next slide.
Done, sir.
When you see the nerve supply of the Nabi is being supplied with T11 and L1 nerves from the spinal, and through this, and there is a direct, because it contains around 10,000 millions of nerves as well as the 7,000 blood vessels, and this is having a direct connection to the brain. Through Nabi, in futuristic way, we can also deliver the molecule to the brain region also by the neural pathway. Next slide. Next slide, please. By seeing all the anatomical features on the nature of the Nabi ideal properties, and this Nabi can be used as a drug delivery platform because of its unique talent features, because it has an excellent site for the application. We can deliver, and drug molecule can be delivered in the suitable formulation. Same molecule can be without any disturbance and for a longer period to elicit the systemic action for the longer duration. Also, it can be used for sustained release. Due to its specific shape, drug-load device can be secured in the place for a prolonged period, and the skin folds of a knobby provide larger surface area because it is having a multiple folds. So it gives a larger surface area where the drug molecule can easily absorb, and by means of diffusion mechanism get the maximum therapeutic action. Of the nabi is the thinnermost body and most part of the body and devoid of the fatty tissue. Nabi area is rich in blood supply, nerve ending, which is closer to the skin, which ensures the systemic delivery of a drug. And Nabi drug delivery loaded devices can use therapeutic management of various diseases. Apart from that, it is very easy to terminate an administration of drug when and wherever necessary. And since it is having a enrichment of a blood vessels, it can be substitute for intravenous. It can mimic the intravenous absorption when you're going to deliver through the. Next slide. Next slide. Next slide, please. Coming to the advantages of transnavi drug delivery, this transnavi drug delivery having unique advantages. Can be administered to the pediatrics, geriatrics, unconscious patients, bedridden patients, psychiatric patients, any normal patients. Patient compliance will be maximum because it is a non-invasive technique. And it is non-invasive, painless, simple, ease of application, reduction of a dose frequency can be done by transnavi delivery. And it is easy of drug administration, and you can improve the bioavailability by various mechanisms, advanced mechanisms by means of using or by improving the particle size and by the particle size and improving the absorption of the drug can be achieved by various techniques. And you can always the side effects because the dose frequency and doses can be minimized through this route so that the drug offering side effects can be significantly minimized. And apart from that, we can also improve the therapeutic level in the blood, therapeutic concentrations of the drug in the blood, and it can be maintained until the dose intervals until the therapeutic expected therapeutic period. Transnavi drug delivery devices offers a flexible termination, as I said earlier, and quickly removed if a person is any action it can be terminated at. Next slide, please. And every drug delivery system, it will have a limitation. Likewise, the transnavi drug delivery is also having a limitation. Navi area is limited, so it is very much feasible for those drugs or the dose because of site specific. This route is not suitable for the delivering a skin API and is not feasible if any person is suffering from Nabi disorder. It can be used only for Nabi treatment, means localized treatment. At that time, the systemic delivery it's not feasible if a person is suffering from Nabi disorder. Next slide, please. Next slide. All these features using a natural function which are being isolated from nature and characterized its potential and developed the formulation films and so that it can be delivered to the liver region through the Navi region, that is called as therapeutic moiety delivery via transnavi. Next slide. Here we have chosen a atorvastatin. Atorvastatin moiety, it is a molecular formula C33H25FN2O5. Molecular weight is 558.64 grams per molecule. The BCS classification class 2, it works by mechanism of action of this atorvastatin is selectively, competitively inhibit the hepatic enzyme HMG-CoA reductase and prevents the conversion of HMG-CoA into mevalonate. Log P value is 0.6 because the site of action of atorvastatin is at the liver, then the molecule is being placed in the Navi region by suitably developing a dosage form. The atorvastatin reaches into the targeted site, that is liver region via trans-Navi route, and the therapeutic moiety will show the action with a minimal dose to the patient by this. Next slide. Next slide. It is adverse reactions like arterialgia, diarrhea, dyspepsia, dyspnea, increased transaminase content, insomnia, limb pain, muscle spasms. These adverse reactions are being reported if a person is going to take orally for a longer period. But this adverse reaction can be minimized, and if the delivery route and the dose can be administered via through the trans-Navi route. Next slide. So for developing a dosage form, first and foremost thing, we require a functional aid, that is the bioflexi films we have developed. For that developing a bioflexi film, we required one polymeric substance, and the polymeric substance should be this films for making. There are so many natural agents are available in the market, and the synthetic polymers that are available. But we isolated a novel biopolymer from the cucumber, the seeds, and by this we isolated the polymer, and then we have seen the filmability of this polymer, the characterization of various unique features. And first we have isolated the polymer by taking the seed and soaking in the water, ground still water, and after that it has been grounded to get in the paste, and it is subjected for the centrifugation by adding water, and the supernatant liquid which has been taken and optimized concentration of propane 2-one was added, and then this solution was kept in the refrigerator for 24 hours, and the biomaterial which has been obtained been subjected for centrifugation in order to collect the biomaterial. The obtained biomaterial has been passed through the sieve and purified this method, and we have calculated and we observed the nature of the material. By this process, optimal process, we can extract around 13% of the polymer from the cucumis mallow seeds, and the texture of the polymer is very smooth, fine, and the color will be light brown color, and the order will be orderless, and the solubility and it is the positive reaction with the carbohydrates as well as the protein, and its color changing point is 185 to 190 degrees centigrade. Since it is a polymer, it will not give the melting. This is also one indication. The color of the polymer will be charring point, that is called as color changing point. That is 185 to 190 degrees. The polymer starts charred. After that, we analyze the functional groups, what are the functional groups that have been present, and what is the GTT value, glass translution temperature, by performing IR as well as DSC. IR study shows that it containing a hydroxyl group, as it shows a special peak at the 3000 area, and which indicates that this is a hydrophilic peak, and which is a hydrophilic group, which is having feasible, and it can be adhered properly when it's been kept in the umbilical region. And its GTT value that showed 90 is by, which was confirmed by DSC study. Next slide. Next slide. Next slide, please. We observed the name of the polymer got under the scanning electron microscope, and we got in a smooth surface with a platy structure. This is a smooth polymeric nature. It has been absorbed by the structure or some atopic been shown in these some pictures. Next slide, please. Next slide. So this once the polymer has been isolated from the cucumis below state, and we have identified whether this polymer is safer. By this polymer study, there is cell line toxicity study was being performed by using neuroblastoma cell lines, and there it showed a duad of a toxicity, and when compared to the molecule, standard molecule, they are taken doxorubicin. That indicates that this polymer is safe and it can be used for the delivery of by using this polymer. Next slide, please. So after getting the polymer, we just formulated the bioflexi film by atorvastatin. First, we have taken atorvastatin. It has been subjected for nanosizing. After nanosizing, the bioflexi films were formulated by taking the polymer and added with the dextrose and loaded with the drug molecule and dissolved in the water. After dissolving, then it has been poured into a petri dish. It has been operation to get in a films. The films are being peeled and cut into different size, 1 centimeter squares, 1 square centimeter, and the big for is the inbuilt parameter. Next slide, please. And we have done 10 polymers, 10 concentration because was taken, and after that we isolated after formulation. We selected the best formulation based on the performance. It has showed the ACM2. It has showed a best polymer, best concentration where you can prepare a bioflexi film that loaded unique functional properties. Next slide, please. Next slide. This polymer has been evaluated for its weight, thickness, surface area, as well as the swelling property, content, water vaporization test, and percentage of drug content. All these polymers have been, all the films has been assessed, it has been depicted, and among that, the ACM2 film showed a very good, unique properties. Next slide. The thickness of this film was 0.5, the best film that 20 thickness polymer. It is 0.35 thickness, 6.47 is the surface pH. It's having a good folding endurance, and its swelling index is around 22%, and the moisture loss is 7.4, and moisture absorption is 14.8, and the drug content was 86.67 with a T50 12.8. T80 is 28.3 hours. This is the outcome of the films of the best film what we have been collected. And this is the in vitro graph, and based on the in vitro graph by comparison T50 and T80, the ACM2 is being selected as the best because increase in the concentration of the polymer does not show any significant change in the T50, T80. That's why ACM2 was being chosen as a best. Next one. And this we are compared with the standard polymers like sodium carboxymethyl cellulose and HPMC, and a significance comparison when compared to HPMC and sodium carboxymethyl polymeric films. Next one. So after that, we have performed the in vivo studies in the rabbits, taking the film and placing in the rabbit navel, and we observed. And here we are choosing a 3 level of dosing. That is the dose, the dose folds and 2000 folds. Response say significant reduction in the LDL, and there will be significant increase in the HDL value. So, to the that is to the rabbits, which has been with the cholesterol-induced rabbits. Hyperlipidemic rabbits were given. This is, and how the therapeutic efficacy it has been shown means. Go to the back slide. The drug might have been shown the pharmacological action via the neural pathway and through the blood vessels. It might have been reached to the liver region, and it elicits the pharmacological action so that 200-fold of the dose can be reduced via transnavial route. So the counseling steps, if the things will come into market in the near future, the patient has to be given a proper counseling for the administration of this dosage form, even though it is a non-invasive dosage form. The patient has to be counseled or educated. First and foremost, the patient's navel area should be cleaned properly, and apart from that, the person has to remove the backing membrane of the bioflexi film, and then he has to place on the fingertip, and then he has to transpose on into the navi area, and he should apply 30 seconds of a pressure so that it can reside in the navi region, so that it can remove words, and the dosage form will be kept in the navi region, and the dosage form starts releasing the therapeutic moiety, and it shows the therapeutic patients continuously as per the desired duration. Next slide, please. So this is the one example we are given, and here the main purpose of this is there are so many biofunctional agents that are being used, and these biofunctional agents have been attained their filmability. These filmable, since they are being having inbuilt filmable property, same functional agent can be used for the transumbilical. So this is a one ideal tree. Ideal tree, what is the main purpose of giving this ideal tree means? Every part of the tree here is used as a biofunctional agent. This here we isolated a biopolymer from the coconut. This is a soybean, cinnamon bark, as well as beetroot, mangoes, and this is the marigold flower. Next slide. So, just I would like to give a glimpses on the smart biofunctional agents for drug delivery via transnavi route. Biofunctional agents are isolated from the natural source. It can serve as a co-processing agent due to its inbuilt property. They are used in developing a drug-loaded dosage form. Next slide, please. And here I would like to talk about one of the polymer. Recently, last year, we got a patent on this polymer, Coccus nucifera. We isolated a biopolymer from the Coccus nucifera. It belongs to a family, and the plant part we used is kernel. And this kernel part we isolated, and we have seen its inbuilt property. It's having a very good filmable property, and the polymer can be used for further delivery of the drug molecule via transnavi route. The same structure of this polymer, it shows a irregular amorphous structure and with a platy nature, smooth structure, granular over the thing. This is the same structure of the Coccus nucifera polymer. Next slide. Next slide, please. Here, in this, we have tried after isolating polymer, we have nanosized the polymer also. Polymer we have nanosized, and we have made the polymer size, particle size to 6.5 jet diameter threshold with a PDI 0.311. Next slide. And we also isolated the polymer from the cinnamon. This is a bark portion we use. It is obtained from the dark and dried bark of Cinnamomum melannicum. It belongs to a family, Lauraceae, and the plant part we used is bark of the cinnamon. This is the same structure of the biofunctional agent Cinnamon planicum. This has showed irregular surface, and even though it is a smooth texture and irregular layered structure has been observed in the stem upon the same study. Next slide, please. And this also we have made a nanosizing. The nanosized polymer, this is showed 252 jet diameter and 0.35 it as a PDI. Next slide. And this polymer, we have explored its inbuilt properties. We have delivered the drug molecule silicylene nanos by formulating a nanosuspension delivery and fluoxamine and ear to brain delivery and duloxetine nanogel ear to brain delivery we are delivered. So this polymer can also serve as a biofunctional agent for delivering APA molecules via trans-Navi route. Next slide, please. Next slide. What polymer we isolate from soybean? The soya glycine max we are used, seeds we are used for isolating polymer. It has shown the group the color of the polymer is like yellow color. This polymer will be isolated by using a chloroform by defatting the soybean powder, and the total time we required for optimization is 4 days, and yield of the polymer we got is 81%, and the solubility of the polymer showed soluble in methanol, and it is a proteinaceous solid content has been confirmed by the various chemical tests. Next slide. And it is the particle shape is spherical. The same structure shows 100 micrometers, and the IC50 value of this polymer on H9C2 cells is 55, and it is a film used for delivering the molecule through soft molecule to brain delivery by loading topiramate into this biopolymer. And it showed 40.6 hours T40 and 44 as a T80, and this has been confirmed by a in vitro diffusion MS diffusion apparatus which has been designed by us. Next slide, please. One more Tegutus petula, which is obtained. This is a pet use biopolymer we isolated by using Tegutus petula, and it belongs to aspect. Please. This is also this petal the polymer after isolate this polymeric is being used for preparing nanogels, bioflexifilms, and since it is having a flexibility property apart from that film, this polymer can also serve for trans-Navi. Right? And the polymer we isolated from Magnifera. It's obtained from Magnifera indica. It belongs to Anacardiaceae family. Here, the pulp portion we have taken by using a non-solvent addition method. We isolated the polymer, characterized, and we have screened for its inbuilt property. Slide. Next slide, please. So, polymer also showed the solubility, and we have tried to deliver the aripiprazole via transcranial route, and escitalopram for ear to brain delivery, lorazepam for transcranial drug delivery, provident bioflexifilms were made for buccal delivery, olanzapine biofilms were prepared by using translabial delivery, risperidone biofilms were prepared by using translabial drug delivery. So this can also serve as a biofunctional agent. Next slide. So on this polymers, what I told, these functions are given by using these functional agents and develop a special type of a dosage form for trans-Nabi delivery route. And these are all some futuristic drug delivery doses where we can deliver a molecule through this route by suitably loading either using a biofunctional agent or a synthetic or a semi-synthetic or semi-natural polymers. One is in situ bio known as sponge. Here we have to means in future it can be developed. If you make a nano sponge on the spot, that is pH sensitive polymer, it has to be taken and preferably biopolymer. And when it is going to apply on the site, it should convert into gel. While application, it should be in the form of liquid. And we can also develop in the form of proneosomal formulation, liposomal gels, nano gels, ML gels, in situ gels, in situ films, suspended integrated lint, nano sprays, nano cream, soft caps, etc. can be developed for this group. And since this is what are the API molecules and can be delivered? This is the placenta. There is a direct connection from there. There is a connection to the liver. There is a heart and the urinary bladder. And not only that, it's having ability to connect to pancreas and pancreas also. So hence, having a connection, and we can deliver a site-specific drug delivery system because of its unique connection. You can deliver the drug delivery to the heart region, the side. You can the urinary bladder and drug delivery to the brain region. As I told, it containing a different billions of nerve endings which directly connect to the brain region. Through the neural pathway, the drug can be delivered to the brain region in near future also. And we can also target it, already we are targeted to the liver. It can be further by delivering through this trans-Nabi route. And pancreatic cancer can also be treated by using this route in a very smart way. And delivery to the pancreas, you can deliver to the adrenal gland, deliver to the intestine and colon, etc. Of this Nabi route. Slide, please. These are all the references. Slide. Yes, these are all the outcomes of research. And I would like to thank to our research tutors, Dr. Harish Chandra, who did PhD under my guidance, Dr. Keith Singh, Bhavna Singh, and Dr. Sugandha for this work and by isolating the biofunctional agent. Next slide, please. I'd like to acknowledge DIT University for providing a and for executing our projects. And I would like to thank medical therapists for to deliver this talk. And last, I would like to thanks very much thanks to Gurukul Kanti Vishwavishwalaya for providing this platform to deliver this talk to the audience on this auspicious day. Thank you.
Thank you, Dr. Satish. Session open for discussion and question and answers. If audiences have any questions, they can ask to our speaker here. Any questions from audiences?
Any question from your side, participant? Any participant? I think there is, madam, you can proceed.
Yeah. So, sir, I would like to ask you one question. Dr. Satish?
Hello.
Yes. Yes, sir. I want to
Yeah.
ask you one
Tell me.
question. Yeah. Sir, have you performed
Yeah.
any studies? Have you performed any studies on biodistribution through trans-Nabi delivery? That you know how a drug, when it is delivered through how you know it distributes across the various organs in the body.
Here, it will not cross the various organs because body, all the organs are being developed through this route only. And this is called as a Sira Marma vessels. When you're going to place the molecule into Nabi region, it is going to take away and it is going to circulate to the particular region. It's having a special because when a drug is being delivered to the particular, they'll be having a specific receptors, the drug will reach and once it enters to the portal or through this route.
Okay. So I think sir, it'll get into the
So,
Okay, I am audible. I want to speak something.
Yes, sir. Yes, sir.
Is it my voice is clear?
Yes, sir. Yes, sir. Your voice is clear, sir.
Okay. Okay.
Yes, sir. Clear, sir. Yeah.
Okay. Dr. Negi, I'm just taking. Actually, there is some voice break is there in Dr. Madhav. So, may I proceed? I want to say something about this talk.
Yeah, sir. Sure. Yes, sir. I would definitely request you to please conclude the session and obviously.
Okay, so Dr. Madhav is a very pioneer researcher, you know, in India and also in maybe in this part of the globe, who is working on the biopolymers, being in in a pharmaceutics. It's not audible.
Yeah.
Yeah.
So being in a pharmaceutics field, he has exploited the biopolymer in a very big way for delivering the drug to different sites, especially on the oral sites and about oral sites, including the brain, exploiting the different types of the biopolymers from the different plant sources which he was just mentioning. But now what he's working in Nabi, I think he has got some inspiration from maybe godly inspiration, because Nabi is an area of the body which really gives all, our body, Nabi is your center of whole body control. As you have already indicated that many of the nerves, blood vessels and so many things are, are getting connected with the Nabi, and the whole body organs and everything is getting controlled through the Nabi. And that's why when child is in the womb, in the uterus, before birth, it is only through the placental blood who is getting completely nourished and getting all his systems developed. So therefore, I am really wondering that through the Nabi, we already know that just you have a lap of a hing at the Nabi area, and you will be completely relieved from the abdominal discomfort or abdominal pain. And it is having so much of the blood vasculature, and we indicated in his presentation that how by a biofilm of a atorvastatin has reduced the dose of atorvastatin to 200 times, and has resulted still into a very good lowering down of the lipid profile and also increasing in the SDL level. That is a wonderful observation, and that is a clear indicative of that how the Nabi can be widely utilized as the external surface as a for topical application, but for getting the systemic use of the drug, for the systemic treatment for anywhere in the body. As far as your question is concerned, has he done, Dr. Poonam, which you asked about any biodistribution studies? Actually, that was also in my mind, this kind of question, because the future prospect which he has just spoken about, the targeting of drugs can happen through this route, through the different organs, heart, liver, etc., etc. But there is still, I have to add into this, that the drug through the Navi is going into the blood circulation, will be reaching to the different organs, no doubt about this. But how to have the localized delivery at a particular organ, that need to be still be worked upon, and that may be based on the same principles as we are doing in the nano formulations or other the targeted drug delivery systems. So that is something which is really good. But the one thing is clear. I think Dr. Madhav, you could also focus that I think this route can completely avoid the first pass metabolism. Many of the drugs, are getting destroyed and is getting so that.
No, sir. Actually, this
first
one,
pass
it is
metabolism can
used for
completely.
delivery. Yeah, and it can be delivered to the liver also, sir. Liver cancer, because it has a special venous supply to the liver, we can easily target to the liver cancer in future.
No,
And we are trying
my concern is that there is a direct vein from the Navi reaching to the liver, or it is going to the systemic route?
No. There is a direct vein reaching to the liver.
That means the first pass metabolic drug cannot be a good candidate for this particular.
It cannot be a good candidate. Yes, sir. It cannot be a good candidate.
Okay. That was my actually concern. So you have now clarified it that they cannot be a good candidate.
Yes.
But anyway, I fully agree with this, that the Navi can be utilized as a very, very, promising place in the body, that too on the external surface, to be applied locally. And the most, beautiful aspect of this delivery route may be that if anybody is feeling any kind of the side effects or the toxicity, that film can be removed as they. It can be, drug input can be terminated. But my concern is that how to really restrict the formulation to remain attached with the Navi, because it is an uneven surface, and you have though indicated with your that one protocol that how it can be attached to the Navi, but that can be, of course, be taken care of, and we can.
It should be properly adhesive, and you can put a surgical tape over the Navi, so that it can be retained for longer time.
No, so that is the concern actually, because by putting the surgical tape or any other adhesive material, they may not be good for long term. They may not be good for the long term use. So that also.
Yes, sir.
There also there has to be something to be done in the way that, like as you talk about the many in situ applications, but those in situ applications, that's fine, in situ gel, but they should be staying properly with some kind of the bioadhesive property. So that is my way of thinking. But anyway,
pro-neosomal like pro-neosomal and need not go for hydration. It will in situ hydration. It will take. So that can be.
Yes. So thank you, Dr. Madhav. It was really very interesting, and we have opened a platform, Navi, as a site for the drug delivery, and I hope many other researchers will take the lesson from this and will learn something and will utilize, including me.
It's a pleasure, sir.
And initially, I really enjoyed your talk, because you started with the anatomical structure of the Navi and how it is connected with the nerves and the vessels. This is something which is, is a good indicator of a good scientist and good orator and good academician and researcher. So that is what you try to explain right from the beginning, that how this Navi can be utilized as a drug delivery center, and then you came to your own. So I really appreciate, and thank you so much for delivering such a wonderful lecture. And I think the audience must have certainly been very, very benefited with this kind of talk which you have delivered. And I really thank you from my own behalf and also from on behalf of the organizing committee, which they may be doing. But I am really privileged to be the chairing this session for your talk. Thank you so much.
Thank you, sir. Thank you, sir. Thank you for your
Uh,
kind words.
thank you, Professor Mishra and Professor Madhav. Now I would like to thank the chairperson and the speaker and the audiences. Over to Dr. Abhishek.
Thank you, ma'am. Good evening, all. On behalf of Department of Pharmaceutical Sciences, Gurukul Kangdi, Vishwavidyalaya Haridwar, I, Dr. Abhishek Bansal, feel honored and privileged to get the opportunity to propose vote of thanks. I convey my regards and hearty thanks to our eminent speaker of the day, Dr. N.V. Satish Madhav, Head and Research Development, Vital Therapeutics, Hyderabad, who spent his busiest time for gracing the session. Today we had the opportunity to hear you, sir. The session was very informative and wonderful. Thank you for adoring the session. We are also thankful to our chairman of the day, Professor B. Mishra, sir, Professor in Pharmaceutics, BHU, and moderator, Dr. Poonam Negi, ma'am, Shulni University, for accepting our invitation and giving their valuable time. A special thanks to our principal, sir, Professor S.K. Rajput, sir, for his continuous motivation and support. We also thankful to our participants, teaching and non-teaching staff for their active participation. Thank you once again. Thank you for giving us time.
Thank you, sir. Thank you.
Thank you, madam.
Thank you, sir.
Thank you, sir.
Thank you.
So we can leave, Abhishek?
Yes, sir. Yes, sir. We can leave.
Okay. Thank you.
Okay.